Good morning, everyone. Let me start my video here. This is Ranjit Riar from California State University, Fresno. And my co-chair is Gina Colfer. So Gina, if you can go ahead and introduce yourself, please.
Hi, good morning. I'm with Wilbur Ellis on the Central Coast of California, and I'm here to help moderate with Ranjit on the weed management portion of this meeting.
Perfect. So today we have this session on current, first, welcome to the second day of California chapter of American Society of Agronomy annual California Plant and Soil Conference. So this session is on current issues and advancements in weed management. And as you all know, as a grower or a researcher, that no matter what crop you grow, or even if you rotate your crops, you are fed up with weeds in one field. In one crop, you try to rotate to another crop, you don't get rid of the weeds that quickly. So it's a long-term fight between the growers and the weeds. So there should be sustainable and long-term approaches that keep those weeds under check. So in today's session, we have three presentations. So the first one is the increasing prevalence of palmer amaranth and common water hemp in the Central Valley. And the speaker is Dr. Anil Shreshta. So Dr. Shreshta is a PhD in crop ecology, and he is a professor in weed ecology at California State University and is also the chair of the Viticulture and Enology department. So with that, I'll turn over to Dr. Shrestha for his talk on the increasing prevalence of palmer amaranth and common water hemp in the Central Valley.
Okay, good morning. And first of all, I'd like to thank the organizers and Ranjit for inviting me to share some of our experience with palmer amaranth and water hemp in the Central Valley. And I would also like to acknowledge my co-author, Dr. Catherine Wasselkov, who is in the Department of Biology here at Fresno State. So these two species, I thought they were the same for a long time, but didn't realize that they were two different species because palmer amaranth is more common around here than water hemp. But both of them belong to the Amaranthaceae family, and they look similar. So there are about 38 species of Amaranthus in the US and 75 species worldwide. It's a summer annual dicot, and it's a C4 plant, both of these species. And actually, these are both classified among the top 5 worst weeds in the US agriculture by the Weed Science Society of America. I think they rank number 1 and number 2, if I'm not mistaken. So the other bad thing about these two are they grow very rapidly and up to 1 to 2 inches a day. So that's a pretty rapid growth there. So as I said, both of these species kind of look similar, but there are some differences that I will go through. Palmer amaranth, these are erect plants, and they can grow up to 6 to 8 feet tall, sometimes even up to 10 feet tall. I've seen them up to 10 feet tall, even in vineyards in the Central Valley. The leaves are wider and ovate compared to the common water hemp. And often they have the silvery watermark, as you can see here. Water hemp doesn't have that. And the stems are hairless. These are actually native to the Southwest United States. And you can see that they are native to this area here. And in recent years, it's started to move to the Central Valley. So although it's native to the Southwest, it was confined to these regions, and now it's moving to the Central Valley. But it has moved to other parts of the US, and it's taken over many of these corn soybean systems. And the dates here indicate where when they were first found to be resistant to herbicides. So in our case, it was 2015. This plant has, you could see it on and off on the roadsides and a few plants here and there in orchards, vineyards, and even in annual cropping systems. But in 2015, we started seeing a lot of this plant, Palmer amaranth in this case, in different cropping systems. And Stephen Wright and Sonia Rios were like the first ones to kind of bring this to our attention. And Sonia Rios actually did her thesis on this particular plant. And this is a picture of Palmer amaranth in a vineyard at Fresno State. So water hemp, on the other hand, is, as I said, they look similar to each other, but it's like a little bit more lanceolate than Palmer amaranth, and it doesn't have that silvery watermark. But having said that, it's not always the case that Palmer amaranth will have that silvery watermark. Sometimes they don't. So again, these are also, can grow up to 8 feet tall, and they're very difficult to distinguish from Palmer amaranth in the fields, because they have this kind of inflorescence, the long inflorescence that Palmer amaranth has as well. So Lynn Sosnowski has a good identifying pictures, sets of, like several of these Palmer amaranth and amaranths, Palmer common water hemp, redwood pigweed, they can look similar. So water hemp is kind of new to us, and it's native to Mississippi Valley. There are actually 2 varieties, rudis, which is west of Mississippi, and tuberculatus, which is east of Mississippi, that they are found in. And they were documented to be invading agroecosystems in the 1950s. So we didn't really have much of this water hemp, occasionally here and there in California, but not a lot. But now we are starting to see more and more of this in our systems as well. Rick Miller from Corteva first brought this to my attention way back in 2009 when he found glyphosate escapes of this species in Stockton. So this is water hemp. I thought it was Palmer amaranth at that time, and we did some tests and we didn't really see glyphosate resistance at that time. And mainly it was like the growth stage. If you spray glyphosate late, it's going to escape. But now we're starting to see it more and more, especially in the Merced County area. These are pictures taken from Merced on canal banks and also in cornfields by Jose Carvalho, who is now in the University of Arizona. So both of these are dioecious plants. That means they are separate male and female plants. They can outcross, and traits are passed through pollen. And they're prolific seed producers. Palmer amaranth can produce up to 600,000 seeds, and water hemp even up to 2 million seeds per plant. And this is what the inflorescence looks like. One thing to note is if you feel the inflorescence of a male Palmer amaranth, it will not hurt your hand. But if you feel the female inflorescence of the Palmer amaranth, it's going to be spiky, spiny, and then you can hurt your hand, but not in the case of water hemp. Both male and female inflorescence will not hurt your hand. So if you feel the inflorescence and if you get poked, then it's probably it is a female Palmer amaranth plant. As I said, there are several others that look similar, but you can tell the difference by the inflorescence. This is Palmer, Powell, redroot, smooth pigweed, and water hemp. They all look pretty similar, but there is a difference in their inflorescence. So we started seeing these Palmer amaranth escapes in the valley, and they used to be confined to the edges of the fields, but we started to see them even within the fields. And you can see that Kings County, Tulare, Fresno, we saw them in different cropping systems, including annual cropping systems and perennial cropping systems. But the main issue we saw was in Merced County, where they were invading these Roundup ready corn fields. So we suspected that maybe we have Roundup resistant Palmer amaranth in that area. So, and the reason we suspected this is because there are lots of Palmer amaranth and common water hemp populations throughout the United States that have developed resistance to almost 8 different herbicides, as you can see on this list. And some of them are multiple resistant. So there are populations of, like Palmer amaranth and Waterhemp that have developed to 6, 7 different modes of action. So it wasn't quite surprising to us that maybe we have some cases of resistance here. And we did document Palmer amaranth in this case, sorry, resistant to glyphosate. So you can see the distribution of glyphosate resistant, or in this case, herbicide resistant populations of Palmer amaranth throughout the US, but we have one case here in the state. In the case of Waterhemp, we haven't been able to document any resistance. And in fact, Waterhemp is confined to mostly the central and the eastern part of the United States as far as resistance goes. So we wanted to ask the questions, do we have glyphosate resistant Palmer amaranth and Waterhemp populations in California? Will these species adapt to our local conditions in the Central Valley? And what alternate herbicides do we have? And where did these populations come from? So these are the questions that we started to ask. So yes, we do have a glyphosate resistant Palmer amaranth population in California. Although I say a population, we may have a lot of populations that are already present here. We documented the first case in Merced County, and actually there are 29 states, including California now, with glyphosate resistant Palmer amaranth. Do we have glyphosate resistant Waterhemp populations in California? Not confirmed yet, but I cannot rule it out because we just tested 1 or 2 populations. There are 19 states in the US that have already reported glyphosate resistant Waterhemp. So it may just be a matter of time before we confirm it. We tested different herbicides at the 3-inch stage and 6-inch stage of common Waterhemp, and we were able to control them with the label rates of rimsulfuron, which would be your matrix, glufosinate, which would be your rely, and suppress, which is an organic herbicide. When we sprayed at the 3 inches story, but the 3 inches stage, but the story was different when we sprayed it at the 6-inch stage. Now we started to see, escapes. So we probably don't have resistant, populations in some cases. It may just be a case of, spraying too late. And if you realize that, as I mentioned earlier, these, grow like 1 to 2 inches a day. So you have a very narrow window when you can control them. So, but we are testing more populations, and as I said, it's just a matter of time before we will be able to confirm glyphosate resistant populations, and maybe even multiple resistance in the case of Waterhemp as well. Now the question, the second part of the question is, okay, so we have these species and populations, how well will they adapt to our local conditions? Because we have, if you look at the west side, especially, we have very saline soils, and the whole valley itself is arid. So we wanted to know, can they germinate under drought and salinity stress conditions? So we developed studies to look at these for germination. So we wanted to look at drought tolerance, salinity tolerance, and pH tolerance at germination. So we simulated drought conditions by creating various water potential solutions using polyethylene glycol. And then we simulated salinity conditions by creating solutions of various electrical conductivity using sodium chloride. Now, sodium chloride is not the only salt that causes salinity problems. There are a host of other salts, but we only focused on sodium chloride to keep it simple. And we also simulated soil pH conditions by creating pH buffer solutions. And we tested the germination of both glyphosate resistant and glyphosate susceptible Palmer amaranth and glyphosate susceptible Waterhemp seeds in petri dishes in growth chambers, which are programmed for a constant temperature of around 22 Celsius. That's like the optimum. And we also created these different water potential solutions to simulate drought, and also different electrical conductivity solutions with sodium chloride. And we also looked at the EC, calibrated them with the EC meter to see if they corresponded to what we were aiming for. And we also tested for germination at 5, 6, 7, 8, and 9 pH levels with solutions prepared with citrate buffer and other substances that you can see, other chemicals that you can see on the list. So moving on to results. Here you can see that the germination of both these species look to be identical in the case of drought tolerance. So the germination kind of declines when it reaches negative 0.5 megapascals water potential. That tells me that it's slightly drought tolerant, but not very drought tolerant. Both of these species do need adequate moisture for germination. That's why we probably see it more in irrigated areas. Once they germinate and start growing, then it's a different story. They seem to be more drought tolerant. How much drought tolerant are they while growing? We haven't looked at that yet. Salinity tolerance, as again, is a different story. You can see that they're very tolerant to salt stress. We had germination even at 20 EC, which is very, very high salinity. This is again sodium chloride. And we also saw a difference between the glyphosate resistant and susceptible ones. In the case of Palmer amaranth, the resistant one was actually more tolerant to salt stress during germination. So both of these species are likely going to be seen in these saline areas. And in the case of Palmer amaranth, we even looked at the growth at different salt concentrations, again at sodium chloride simulated concentrations. So you can see that the growth, the biomass is actually reduced only at about 10 EC. And they were still growing even at 20 EC. And we're producing seeds. So these are very tolerant to salt stress and high potential of invading the west side. So you can see the picture here, even at 20 decimeters, they're still growing and they produce seeds. So they're more likely to be invading the west side as well. pH, they seem to like pHs of 7 and above. And again, this makes it a case that they are adapted to higher pH areas, and that occurs in the west side. So what alternate herbicides do we have for post-emergence control? And this is actually results from Sonia, who looked at different growth stages of the Palmer amaranth. And you can see, like, in the case of saflofenicil, which is your Trivix, paraquat, which is your gramoxone, you get good control even up to the 12 to 16 leaf stage. But with glyphosate, glufosinate, and rimsulfuron, as the size of the plant increases, the percent control decreases. So that's why, you got to get them very early. And again, the challenge is these grow like 1 inch a day. So you have a very small window of getting these with post-emergence control herbicides. So she also looked at tank mixes, which I will share. So this is just a picture showing, as you wait longer, you start to see escapes. So she looked at different tank mixes, and she looked at the 8 to 10 leaf stage. So she got really good control with tank mixes of Trivix, Weathermax, Rely, Clarity. As you can see on this list, 100% control. But then be aware that these have developed multiple resistance in other states. So just because we are able to control them with these tank mixes in California at the present situation doesn't mean that we will be able to do so forever. So we got to rotate these chemicals, but we do have a good arsenal of chemicals still working on these two species. We haven't really looked at Waterhemp as far as alternate herbicides go, but I know Rely and Suppress, which is an organic herbicide, both worked. And we plan to continue our studies to look at different herbicides to see if we can get the same results as in the case of Waterhemp. It was interesting to kind of learn that there is genetic controls studies being conducted at the University of Illinois and actually in Davis's well, where they're developing populations. Well, the goal is to modify the male plants that they would mate with female plants and produce nothing but male offspring. So, it was very interesting to hear these kinds of studies going on both in Illinois and Davis. So we might see some of these efforts in the field where we have modified plants mating with females and producing only males. So the last question was, where did these populations come from? We sampled several populations from Merced in almond, corn, hay, wheat, rice fields, and we compared them with Midwest populations with the help of Dr. Wassilkov. And we find that we have both the rudis and the tuberculosis populations here. So, but Merced populations are derived largely from the Western cluster, which is your rudis type. And some have both genes. So they may be moving from other parts of the state. So Palmer amaranth was same. And but we have populations that are native to Southwest, so it's not surprising that they may have moved from the Southwest part of the state to the Central Valley. So our analysis shows that we have both the rudis type and the tuberculosis type in Merced County, but we're still looking at the genomic data for the Waterhemp and as well as Palmer amaranth. And why is it important? Because these may have moved with movement of seeds and hay from other parts of the state. So not only state, but the US as well. So the conclusions we made from these studies is, yes, we do have glyphosate resistant Palmer amaranth populations in the Central Valley. We have not confirmed it yet with Waterhemp, but as I said, it may just be a matter of time. Both seem to be very drought tolerant. At germination, I shouldn't say very, I think moderately tolerant to drought at germination, but very salt tolerant and have the potential to invade the west side with in the areas of pH range of 7 and 9. Best to control these species with post-emergence herbicides before they reach the 8-leaf stage. And there are several options available to us. And we have a mixture of different species, subspecies of Waterhemp in Merced. So both the eastern and the western rudis and the tuberculosis type. GR Palmer, glyphosate resistant Palmer may have come from elsewhere. I suspect with the corns, Roundup Ready corns seeds in Merced, because that's where we first documented them. So the funding for this work was provided by the USDA NIFA AFRI grant to Fresno State and the University of Illinois. And we also got some funds from the CSU ARI system. And also thanks to the Fresno State Horticulture Unit and Jordan Agriculture Research Center and the biology department for letting us do the work. Thank you to colleagues Lynn, Jose, Steve Wright, Sonia Rios, and Brad Hansen. And there were a lot of students who helped with these projects. So Samiksha, Gabriel, Josue, Christine, Takui, Kelsey, Alexander, Romy, Eddie, and I may have even missed some folks. So with that, I'd like to end and thank you for your attention.
Great, thank you, Anil. Oh, if everyone would like to do the DPR poll, so you can get your DPR credits right now before we jump into the Q&A, that would be great.
And we can let that run in the background for a bit if we want to get to that question while people fill it out.
And then Anil, I have a few questions here for you in the Q&A. So the first question, what are some of the challenges that a farmer that uses organic IPM can face when dealing with Palmer amaranth?
Okay, so with organic IPM, if you're using organic herbicides, suppress seems to work. The other thing with these is seed bank management is vital. So if you see Palmer amaranth escapes, let's not wait till they produce seeds, because you see that they can produce like, up to 600,000 seeds. So getting rid of Palmer amaranth, even if it is pulling out by hand, you have to be on top of these, both amaranth and Waterhemp, to prevent reproduction and prevent future infestations. And the second question there is, can cover crops control these weeds? There are studies from the Midwest and Eastern US where they have found success with rye cover crop. And you won't get 100% control, but you will reduce the populations of Palmer amaranth and Waterhemp.
Great, thank you. And I have a quick question also. So I know, a lot of these weeds are also in non-crop areas along highways and the water canals. Are public works working with you, or are organizations that are working with this kind of information to get to them that we have these resistant species out there, and to maybe modify what they're doing? Because I know, right, with each plant setting 600,000 seed per plant, one plant going to seed can cause some major issues.
Yes, fortunately, the Caltrans folks and other non-crop folks do attend these meetings. By these, I mean the California Weed Science Society meetings. And there were several talks on Palmer amaranth and herbicide resistance in general. So they are aware. The problem is that these grow so fast, and we have a very small window of controlling them with post-emergence herbicides. And in roadsides, they use mostly post-emergence herbicides, but in ag settings, we have pre-emergence herbicides, which people have started to forget. Pre-emergence herbicides can prevent the problem a lot. So if you rely just on post-emergence herbicides, you set yourself up for trouble.
Great. I think, do we have one more question here? Oh, so another question. What is the length of time the seed remains viable?
I think with these pigweed seeds, more than 10, 20 years. I don't know exactly, but they have a very long life in the seed bank as well. Unlike your Marystale and Fleabane, which don't live for more than 2-3 years. But these amaranths, they live for a long, long time. Okay, the first question was, Palmer amaranth and common Waterhemp are dioecious plants. That is, they have separate male and female plants. Yes, they have separate male and female plants. Second is Palmer amaranth and common Waterhemp populations are reported to be resistant to more than 5 herbicide modes of action. And that is true. As I said and showed, that there are reports of like 7 or 8 different modes of action now that have cases of resistance in both these species.
Well, great. Well, thank you, Anil. Very, very great presentation. Thank you. So now let's switch gears, and I would like to introduce Pauline Canteneur, who has been with FarmWise for 3.5 years and wears many hats for the company. She's here today to speak about these new automated weeding technologies and how they fit into intensive vegetable farming systems. So thank you, Pauline, for being here today. Take it away.
Hi, thank you so much. Really happy to be here as well. Let me share my screen. All right. So thanks everyone for having me. I know I'm not your typical speaker of the day, as I represent the industry. And I'm also not a researcher or scientist myself. I have a degree in business management, but I hope I can bring some maybe different perspective on all this subject, and something that's maybe commercially available, and discuss state-of-the-art technology. So I would love to tell you a bit more about what we do, why we're in business, the kind of technologies we leverage. I have a video for you as well, so you can see the actuation of the machine, and then we'll have some time for Q&A and some poll questions. So at FarmWise, we are in business to help growers deal with the new societal, environmental, and regulatory challenges that they're facing. And in particular, we want to be here to address the shrinking labor supply. We added a typical, so this slide is usually for venture capital investors who sometimes do not know what's happening in agriculture necessarily, or how, just like a lot of people out there are not really aware how labor intensive specialty crop production is. And so what we're targeting first is the weeding market, particularly the post-emergence weeding passes, folks going across the field, hoeing, with a crew of 15 to 25 people, once or twice per cycle on leafy greens and brassicas, typical crops from the Salinas Valley. So this is what we're focusing on first. As we all know here, weed control is super important. As I like to put it, it's both, a near-term investment and a longer-term investment for the growers. And so we want to be helping with that. And on the other side of the coin, this is one of the most labor-intensive process on the specialty crop farm, along with harvesting. And so we thought it was a very interesting first use case to tackle. And so we brought to market this robot that you see here on this slide. We call it Titan. Titan is not, it's the first commercial robot we bring to market, but it's the third iteration. We've had a very first prototype that was very ugly. Then we had a second generation of machine. You can still see some pictures online. It kind of looked like a tank defense vehicle. And now this kind of looks more like a cabless tractor and what we call a smart cultivation implement. So under the hood, number one, this is where the magic happens, where we have camera sensors and specific light orientation to allow the machine to do its thing. So we tap into deep learning on the one hand so we have really strong vision capability. So contrary to the first generation of smart cultivators that came up, in the year 2010, RoboVator, RoboCrops, TKT, those might be words, company names you've heard or product name you've heard before. This technology uses infrared technology. So to put it very simply, they are able to detect blob of green against dirt, against brown. This works very well in a lot of cases. It can present some challenges in a very weedy field, or when crops are a little bigger, or when crops are planted at a higher density. That's the case with celery and broccoli compared to your lettuce crops. And so on our hand, we're really tapping into neural networks, and we do that in order to assess the perimeter of the crop and as well as the stem. So we're coding an algorithm that not only detect crops from weeds, but also have the capability to really assess where the root zone are probably coming down and where the meristem is located. And that allows, very precise calibration of mechanical tools. That's the second kind of side of the machine. And so to get there, we've been collecting crop images for years. And so it doesn't work like we're just out there collecting images and now we're commercially available. We've been, working with growers hand in hand and like having machines running out in the field and not only validating the vision, but also the tools at the same time, which is, always extremely challenging. But we've been collecting like beyond 300 millions of images of crop. Now, it doesn't mean that we annotated every single image we collected, because that's also very costly. We have selection tools that allows us to really find the images that are the crustiest. So what you want to do when you build this kind of models is to expose the system to all the scenario it's going to be actually facing out in the field. So scenarios where there are very little weeds, scenarios where they actually is a huge weed density, weed pressure is really high, weeds look like crops from a distance, and then obviously different kinds of crops. The goal is really to be able to help those specialty crop growers with not necessarily all the commodities, but most of the commodities that they grow. Knowing that there are a huge difference between, carrot crops and a lettuce. The second part of the machine is all the robotic planning. That's how the robot really understands its environment, how it perceives its position versus the rest of the bed, the plants that are around. And so at the same time, while this assessment is made, once the crop detection model, so think facial recognition, but for crops, has done its work, we actuate blades in between the crops. So we open and close blades in front of the crop. And we also have fixed tools that are attached to a cultivation bar so that we clean the entire bed, intra-row and inter-row. And current crop portfolio is written down there. Really, really proud to be able to weed stuff like broccoli and celery because of the tighter spacing that they offer. And I think just sharing the video will probably speak for itself. If it doesn't show extremely well, feel free to email me. My email will be at the end of the presentation, and happy to send it out to you guys. It's not a public video, unfortunately. Each machine is operated by a human operator, an equipment operator. That's for several reasons. One is quality supervision and improvement of the machine product iteration. And this is also California regulation today. We have not applied for an exemption on the law that doesn't really allow today autonomous farm equipment to be out there in the field freely. So here you can see the little blades that open and close. That's the machine going pretty fast. We do close to an acre an hour on lettuce today. That's that naughty purslane just covering that field. And here again, so here you can see this little blue pipe. We've had several iterations of how do we make sure we're not throwing dirt on the crop, and how do we keep the crop pristine, because we obviously work for fresh market vegetables as well. And so we've had the system of blowing air. It's not there anymore, but we've had that in the past. That's broccoli, and you see the blades are a little different. So we custom design all our blades. That's kind of the second part of the machine, right? We're both a vision system, and we're also, we're moving dirt as we go. So there is iteration and refinement that goes into the tooling that we leverage. So it doesn't mean that each crop has its specific set of blades, but it's a combination of soil conditions, field conditions, spacing in between the crop. It's very weedy cauliflower down in Santa Maria, and also crop. So we've designed close to 200 different types of blades. We're actively using 15 of them. That's how it works. And I think that's just nice drone shots, but nothing that you've not seen so far. All right. Okay, so a few words about the way we deliver this product today. So again, Farmers is commercially available. We've been generating revenue since 2019. And today we have 14 machines of the generation that you saw in the video. And we operate as a service provider. So just the same way the growers call Wilbur Ellis or Nutrient to do custom, pest control applications, they will call us to take care of, some portion of the weed control program on some of their acres. And so the chain of command is really simple. People call us, say, hey, I have 15 acres of celery for you to weed tomorrow, or, by the end of the week. We take care of the whole thing, where we have our own equipment operator driving the machine to the field, operating the equipment, bringing it back to shop. And we have 2 facilities down in Salinas, and from Salinas we cover Oxnard. We're doing quite a bit of celery acres down there. And the beauty, and also I would say like what's happening in the market today in terms of labor constraint is we are now entering conventional celery acres, when in Salinas we're still doing only organic. So I think, Oxnard is facing a little bit more of a stronger pain, let's say, than Salinas. So it's interesting to see how the market geographically shifts and starts, shifting as we go. Why do we operate as a service, and why don't we sell the machines? I think in the future, we want to be able to accommodate that, because we know growers love to own. As a young company, especially like we were complete outsiders, it's been very, very, very important to build that trust with growers. Today, we work with 20 different farms, from Salinas down to Yuma. And this happens with like trial and errors and showing up regularly and making improvements, hearing growers' feedback and building up on that. So we needed that precious, I'd say, field time, constant, steady, regular. And so service allows that, and it allows us to really control the whole process without asking the grower to bear any risk. And it's pretty flexible for them. They can hop on, hop off. So that's usually very easy for us to get work. Now, again, is this the only answer to a successful weeding business, automated weeding business? Probably not. But for now, that's how we operate. And I would say our flagship crops are romaine, broccoli, celery, and in Oxnard now we're doing quite a bit of kale. And we'd love to add, smaller kind of programs like fennel, bok choy, just to be able to accommodate, the portfolio of our own growers. I always like to say a few words about the data that we collect. In order to do what we do out in the field every day, we need to collect certain data points. First of all, each crop is associated with a crop number or plant ID in our system. To this number is associated GPS coordinates, and we're also taking individual pictures of the crop. And so we're able to approximate the diameter of each crop at weeding stage when we do a pass in the field. So really, let's say 2 weeks post-transplanting, 3 to 4 weeks after planting, we're able to offer this kind of like, okay, this crop ID number 3 is has a 4-inch diameter. We have the capacity to assess volumetric data, and we're also measuring the spacing from stem to stem. So that's of interest, especially in a directly seeded field, to be able to say the spacing was maintained. If there was an issue with the planter, that's a way to see it very clearly. We're also able to account for misses, like able to see where plants should have been and where it's not. So there are I'd say, like a few simple metrics: size distribution, spacing distribution, plant counts, GPS coordinate, photo of each crop, and so on at weeding stage. It's very simple, and we're able to give that back to growers. So we built a beta version. That's the report part of my slide. We built a beta version, really early platform, where we're giving those data back a few days after we come in to weed, to a few selected customers in order to see if there were, interest to get those data. And so today where we are at is we realize that, yes, this data has power, but the power comes from numbers, and be able to collect all of this data on all the acres of one program. It's not very useful if I'm giving data on 5% of their iceberg acres. What would be nice in terms of looking at trends across many different fields would be to have, obviously the entire portfolio on there. We're also collecting data at weeding stage. It'd be really great to have several moments of data collection throughout the cycle. So those are things that we're aware and very interested in diving deeper into, and see how, we can maybe empower PCA or help, ranch managers have a little bit more, kind of accurate data, ground truth data to back certain decisions. And then the first two part, stuff that we kind of never really talk about. And I thought it was a mistake, because we it's kind of interesting. We do build a lot of internal software tools to be able to operate our service. So as you see, we have this, fancy machine. And at the same time, we have an agricultural service business, which are two radically different things. And when you have only two robots, kind of easy to know where they are at all times, what they need, do they need maintenance, where they're going tomorrow, what configuration should they have, are we doing 5-line iceberg, are we doing 6-line romaine? When you have 14 machines, things start getting a bit complicated. And when you have, a team of, let's say, 35, technicians, operators, like people need to know what's going on at all time. Especially when you're doing several hundred acres a week of weeding. So we've built this online scheduling tool that kind of allows to track where is each robot at all time. It's a way to kind of say, okay, we need to put this set of blade tomorrow. This robot goes through this field. This is this operator. It needs to be configured to X amount of line. We need to remove weeders and so on. So that's kind of like scheduling. And then we also have a performance tracking. So we're tracking speed in real time. Obviously, so everything that relates to the performance of the robot itself is monitored at all time. And those metrics are, analyzed, looked into, and allows you to debug the machine. And then in real time, we also monitor the detection. That's what you see moving in the monitor part here. We have a team of teleoperation folks who are sitting really in the middle of, in between the field operators, so the guys who are in the field with a tablet operating the robot, and the engineers. So to spare some time to the engineers who are also working on R&D projects, those teleops people are here to kind of pull out diagnostic when the robot needs support or the operator needs support. And so they're monitoring the detection models at all time, making sure we're not picking up nuts edge for whatever crop we're weeding, celery, or that lem's quarter is not a romaine crop for sure. So like, why are we picking it up with this model? And then informing the engineering team to do additional work if needed. That's kind of the data component, kind of the back behind the curtain kind of scene at FarmWise. And just to finish, I wanted also to give a few words about what's coming up for us this year. So we've been iterating and for 2 years with many different growers from Salinas Valley down to Oxnard, Imperial, Yuma. And we're kind of excited about this new generation of product that's coming up. We really think it's going to bring something different compared to what's out there in terms of modularity, so that precision of actuation. And in terms of ease of operation, we don't want people with a degree to operate that machine. We want to be able to hand this machine off to growers and have their own tractor driver operate the equipment. So how do we make that experience really smooth and really simple? Probably through monitor in the cab, which will allow them to really see what's going on at all time. And then reliability. I think that's something that farm robotics company have been struggling with. We're not, tractor manufacturer. We've been making farm robots for 5 years. So being able to offer that kind of industry standard kind of reliability is something that, we fight up against every day. So that's kind of our goal with this new product. And so we're receiving it on Valentine's Day. How amazing is that? And we're going to be doing some testing for the next few months. And we hope to start piloting with growers this summer already in Salinas. And then as we enter the winter, being able to also collect feedback in the desert. And we will start taking pre-orders for this machine very soon. This is a few people within our team. Same thing, this slide was made for an investor meeting. So that's kind of like the way we like to separate all our engineering department, from machine learning to robotics, software engineering, mechanical, electrical engineering. So there is a good split at FarmWise between people who have a strong agricultural background and folks who have never worked in agriculture before, but are really, machine learning experts. Today, we're a team of 75 people, if I keep my counts to date. again, good split between engineering and operations. And that's the beauty. And that's also the challenge, obviously, of the organization, is making sure that all departments can communicate with one another very fluidly, that, engineering language is translated into non-technical language, making sure that, engineers are not favored over agricultural, people. So all of that is really, as a culture, it's super important, and yes, something we work on every day, and it's pretty humbling. So with that, I like to thank you for your attention, and happy to take any question you may have.
Thank you very much, Pauline. So for the viewers, there is a poll that's open now. So please go ahead and finish that poll for the DPR points. And I think we do have a couple minutes for questions. So I'm gonna read it for you. So the first question, Pauline, that we have for you is that, are you able to work in Los Baños, and how much acreage is the minimum required for you to work with?
Yes, so it's not about the minimum amount of acreage because we operate as a service today. I think it's important for us to have steady, regular work in the area with multiple growers, even if your acreage is not, if you're not a 5,000 acre farm, doesn't really matter to us. It's more like, is it worth it for us to allocate a machine down there? We were doing work in Santa Maria for years with one customer, and it only becomes worth it, as we, try to reach profitability with a company, to be able to work with several people down there. Now today, as of this year, I'd say we are in limited capacity. So we're focusing primarily on the regions I mentioned during the presentation. We always would, we're always happy to show the machine, come down for demos, to show you the capabilities, limitation of the equipment, and also kind of plant the seed for the future. But we will be bringing more machines out of manufacturing, out of production, and we'll have machine for purchase very soon. So feel free to reach out to me, and we can schedule some field demonstrations and so forth.
Okay. Right, I think we are out of time. Thank you very much, Pauline. The remaining questions, maybe if you would be able to answer those offline, that would be great for our viewers. So with that, I'll turn it over to Gina. Thank you.
Thank you.
Actually, before we do that, could we make sure to address the DPR questions and just to get the correct answers for the people interested in those?
Yeah. So with the farm waste system, crops are mistaken for weeds less than 0.4% of the time. And our farm waste machines using the same actuation tools blade in any given field? No, we're changing every time. Not every time, but depending on soil conditions, crop density, and weed density, and we'll change the blades. Thank you.
Great, thank you, Pauline. And now I would like to introduce Ranjit Riar to speak next. And he's an assistant professor of agronomy teaching in the plant sciences department at Fresno State. His current research includes salinity management in cotton and alfalfa, fava bean as a cover crop, nitrogen management in garbanzos, and range management. His presentation today will focus on weed management in irrigated pastures.
Thank you, Gina. Perfect. So my presentation for today is evaluation of Roundup for common rush control in irrigated pastures using a Wick wiper. So let's get started. Common rush is a plant. It's a rush. It's not in the grass family. It looks like a grass. If you are driving by any irrigated pastures and you see these tall clumps, like 3 feet tall clumps of dark green grass-like structures, and you think this pasture is really productive, has a lot of forage in it. I had that impression when I drove by the UAL, the University Ag Lab at Fresno State campus, the first time in summer of 2018 when I moved here, and I thought, oh, this looks really good. And then I started looking into it and asked and found out that it's actually a weed. So I start researching it. So it's a very common weed in irrigated pastures, also known as soft rush. And there is another name that's commonly used here in the Central Valley, but I didn't find any mention of it in the literature. It's called as wireweed. So I started searching with the wireweed. I couldn't find anything, so I had to improvise. So this plant forms very large, like 3 feet wide, 2 to 3 feet wide, dark green clumps, and can grow up to 3 feet in height. If the stand density is low, then cattle will move around and graze from around it. But if the density of these clumps is very high, then the cattle would avoid those areas of the pasture altogether. So this weed likes moist or humid conditions. So established stands can be found in irrigation canals, moist or the wet areas, and even it's used as a landscaping or an ornamental plant in certain landscaping plans. So in canals or in the swampy area or in the moist areas of water bodies or channels, any of these established plants, they will act as a seed source. So the seeds, they will be shed into that moving water, or with rain, they will be carried into a moving water source. And from there, as that water is channelized to canals, and it goes into the pastures, and that's where these seeds gets established. And that's how preliminary or the first introductions are made, not through well water, but only in pastures irrigated with canal water. That's where it's a problem. So the seeds, they can be dispersed by wind and water. And once they are shed in the soil, they can live for more than 60 years. 60 years is how long they have been documented that they have remained viable, but they can live longer than that. So here's what the pasture at the UL looks like, or looked like, before it was treated, or the day it was treated with the Wick Viper. So let me give you a premise of why we are using this Wick Viper, and I'll explain here in a minute as well. So this technology is where you are using the height differential in a third dimension, giving you the option to use a non-selective herbicide without spraying it on the crop, or without using any spray hoods, or in a crop that's not planted in rows. So instead of spatial or the sidewise two-dimensional space, you are using the third dimension of height. So wherever the weed is taller than the crop, you can move this Wick Viper above the crop without getting in touch with the crop plants, but treat only the weeds. So here you can see some of these forage grasses, they are taller or as tall as the wireweed or the common rush clump. So these will die because of Roundup. But wherever you have foliage, forages that are shorter and the weeds are taller, the Wick Viper will apply the herbicide only to the tall weeds. And you can see how dense these stands of common rush are. So cattle will graze in these open areas, but they won't go into these closer areas. So infestation of wireweed in irrigated pastures reduces the quantity as well as quality of forage that's available. Young plants, they can be grazed when they are the same height as the forage grasses. But as the plants mature, they have a higher lignin content, and that's why cattle do not feed on these. Sheep might browse on these a little bit, but still it can persist. So you cannot rely on the animals taking care of it once it reaches a certain age and height. It also produces forage productivity due to competition and reduces the forage carrying capacity. So although this weed has other ecological applications, like in landscapes, in irrigated pastures, it's considered as a weed because it has no economic value. So some management strategies that we can think about: tillage, burning, mowing, and reseeding, they are not recommended. So remember, these seeds remain viable for 60 years. So even if you plow it, or if you burn it down with a herbicide or with fire, the remaining seeds that are present in the soil, they will keep on coming back. And even if you reseed, you will have some regermination of those seeds that are already present in the soil. So over time, if you have an established wireweed infestation, you definitely have a large seed bank. So due to that large seed bank, reseeding or tillage won't work. And even if you try to do that, let's say you irrigate once with canal water in case of during the dry summer, when you don't have well water available, and that canal water being contaminated with the seeds of wireweed, all the effort that you have done is down the drain. So this study, the purpose of this study is that as the populations, they tend to build up over years due to seed dispersal from existing stands and from new introductions or repeated introductions with canal irrigation water, the populations build up. The small germinating seedlings, they can be grazed and knocked down by cattle because they have lower lignin content. But when they increase a certain stage of height and maturity, then the cattle don't graze on it, and it also scares them away because it acts as a hiding ground for predators and other rodents. Now, as a herbicide, Roundup can be used, but if you spray it, there is no effective way to avoid the drift. So if you have a backpack sprayer and you have a 50-acre pastures, you can well imagine how long it's going to take. And even a few small droplets of the Roundup solution bouncing off, they will kill all the grasses wherever they land. So how do you manage common rush or wireweed? If you let it be, it will continue to grow and your pasture will continue to decline. So it reduces the pasture health as well as productivity. You can use grazing as a component of your weed management because young seedlings, they can be grazed and it will knock it back, but they still keep on regrowing depending on the grazing intensity and the degree of defoliation. So established plants, they are not grazed and they act as a seed source. So wherever they are, the seeds will fall down. With moving irrigation water, they will move away. The birds and the animals, they will carry it away from one place to another. So Roundup is very effective, but the application is limited because you cannot spray it without causing some damage to the plants around it. So that's where the Wick Viper comes into play. So this is a paint roller strategy. I don't know if many farmers here in the valley have seen or heard about it, but it's very commonly used tool in the Midwestern and in the Southeast, especially in cotton defoliation, where you have the crop where you have to defoliate, but there are tall weeds. So to kill those weeds before harvesting, this strategy can be used. So you have a non-selective herbicide, very high concentration, and you roll it over the crop canopy without getting in touch with the crop, but only treat the weeds. So in this situation, it can be pulled behind an ATV or a small tractor, and there is a spray tank that has the herbicide solution, a pump that pushes. It's a battery operated pump that you regulate. So you will turn the switch on, leave it on for 10 seconds. That will coat the wick cloth on a rotating cylinder with the herbicide solution. And depending on the foliage density of the weeds, you will have to prime it more often or less often. So the herbicide is transferred from that rotating drum to the weed surface as the Wick Viper moves forward. So here's a picture of that Wick Viper behind an ATV. So it's about an 8 feet wide swath. And here is a detailed picture. So these two wheels, one of these wheels has a pulley and that has a belt on it. And this is the rotating drum or the wick, actual wick. That will transfer the herbicide to the weed it comes in contact with. So under this hood, there is a boom of spray nozzles. And here's the spray tank, here's the pump, here's the power cable. So when this pump is primed, a hose will carry the spray solution to the nozzles, and those nozzles will moisten this blanket-like or a wick cloth on this rotating drum that spins counterclockwise. So as the wick moves forward, the belt has a crisscross, so it crisscrosses, and this wick moves in the opposite direction. And it moves as the wiper is pulled forward over the foliage. This will spin backwards, and it will transfer the herbicide solution onto the weed foliage that it is in contact with. A little bit about Roundup. It's a fairly common systemic herbicide that is non-selective in nature, but it's recommended to be applied to a weed that is actively growing. If a weed is under some kind of stress or it's dry, it's not actively growing, then the herbicide efficacy is reduced. So you need to understand the biology of the weed, the efficacy or the mechanism of the herbicide, and the application technology and the strategy to get good utility out of this system. So Roundup and Wick, using a Wick Viper is a better option than spraying because it avoids any off-target movement. So since Roundup is a systemic herbicide, over time it can kill large clumps of wireweed. Typically, Roundup takes about 14 days, anywhere up 10 to 14 days to kill a grass plant completely or a grass weed completely. But that time is much longer for common rush. It takes longer time, but it does kill those large clumps, and they will not regrow from that same area. So the study was designed to evaluate different rates of glyphosate for efficacy against common rush. Applied in spring or early summer when the weeds are actively growing, typically after the first irrigation or warming up after adequate rainfall. And the other objective was to evaluate a single or two passes at different herbicide concentrations to see its efficacy against weed control. So a little bit about the experimental setup. So it was done in 2019 and 2020 growing seasons during the summer. So in the first year, we tested 3 concentrations. 50% is the recommended or the labeled rate. So we tried to see if we can go lower or higher. If higher would give us better control, and lower would save us the cost on the herbicide, especially this year. Based on those results in 2020, we did not test the 75 because it didn't make a lot of difference between 75 and 50%. So there was no point in using the extra herbicide, but we did add a second pass to see if at 25% using 2 passes versus 50% at a single pass, can we save the herbicide versus time, or are we better off using a single pass with the labeled rate? So we looked at chlorosis, which is the yellowing of the foliage, also necrosis, which is the dead foliage as a percentage, and weed mortality at 3, 7, 14, and 21 days after treatment. And then we looked at those plots again at 6 and around a year later after those treatments. So results table from 2019. So here we have the days after treatment, 15, 16, 30, and 60 days after the single application of the Wick Viper, of the Roundup using Wick Viper. And here we are looking at chlorosis or yellowing for 25, 50, and 75% concentration. So yellowing starts at 15% at a lower rate as compared to the higher concentrations. But over time, by about 15 days, it had reached its peak and then started to go down because necrosis would pick up. So this is what we are interested in. That's an indicator of mortality. Yellowing is that the chlorophyll is degrading. Necrosis is when the tissue has died. So over time, you can see there is an increase in necrosis or mortality for all the treatments. But in case of the lower concentration of 25%, it was subpar. But both the concentrations of 50% and 75%, they got really good control, complete kill of the treated plants at 6 months. So at 2 months, there was still a little bit of green foliage left or yellow areas left, but at 6 months, the entire clump had died. So What's the necrosis at 16 days when you would expect Roundup treated plants to or weeds to die? It had only about 20 to 30% necrotic areas. So very slow to die, but it eventually dies as a result of Roundup application. Similar results were found in 2020, so it's a different format, but we are looking at foliage chlorosis, and we did have a second treatment of single pass versus double pass. So the blues are the 50% two passes, the yellows are the 50% single pass, the gray 25% two passes, and the orange is 25% concentration with the single pass, and these are the controls. So looking at necrosis as a result of these treatments, at 30 days, we had only about 60% necrosis in 25% concentration two passes versus about 25% necrosis. At 50%, it was 95%. At 60 days, the 50% single pass had reached, just like the previous year, complete control, and that was at 180 days or 6 months as well. So over time, generally from 2 months to 6 months, you will get a complete kill of these clumps as a result of treatment with single pass of 50% solution of Roundup. So necrosis is slow to develop in wireweed compared to other grasses, but it eventually dies. And this is due to the differences in biology. It's not a grass, it's a rush. And it does have underground storage structures as well, very small, but they do have those haplocombs or comb-like structures. But Roundup is effective in killing it due to its systemic nature. So higher concentration of 75% did not provide any better control than 50%, while 25% provided only partial control, did not result in complete kill of the clumps. Only parts of the clumps were killed. Looks like the lower concentration tends to get diluted and runs out of juice before the entire clump is dead. In the second year, we found that the double pass did not increase the efficacy, either at 25% or at 50%, and it took the double amount of time and fuel. So you are better off using a single pass at 50%. So a single pass application with 50% Roundup solution using 1% AMS, ammonium sulfate, as a tank mix applied to actively growing weeds in late spring or early summer, when the weeds are actively growing, typically kill the clumps in about 6 months. You would start seeing good results in 2 months, but with weathering and total kill, it will take about 6 months for those results to be very evident. Now, AMS or ammonium sulfate, it's recommended in areas where the water hardness is unknown, or you don't know how hard your carrier water is. So as a cheap insurance, it's better to add that in the tank with some water and mix it before you add Roundup. It maintains its efficacy. Otherwise, if there is any hard water, it will bind the active ingredient glyphosate in Roundup, and it will reduce the efficacy of weed control. So 6 months later, when the rain come in the spring and in the winter, or there is repeated irrigations and there's cattle movement over these dead clumps, they begin to disintegrate on their own. So you don't have to mow them or do anything after they are dead. So once they begin to die down, the forages will begin to take over, and they will reclaim those patches where wireweed once flourished. Once those wireweed clumps die, clovers and other grasses, they will resume their growth. So I'll have some pictures in a minute. Some recommendations for successfully using this strategy. Depending on the foliage density of the weeds, you would need to learn how much and how frequently to run the pump. Because if you run the pump too much, the herbicide solution will start dripping after saturating the wick surface or the wool-like structure on the rotating drum. And once that dripping begins, all those areas where that wick viper will move, the dripping herbicide will kill the grasses on the ground as it lands on the foliage of those grasses, because again of its systemic nature and very high concentration. They will regrow, but they will die at least from that initial exposure. So if you have very high density, maybe run it for 5 seconds and travel 10-15 yards and run it again. If you have a low density, run it for 5 seconds or as long as it takes to saturate it, but travel a longer distance before priming that pump again. So once you treat your large clumps and they are killed, you should monitor your pasture for new weed emergence. And based on that, the density of the new weeds that are emerging, you can tweak your grazing pressure and grazing intensity to push the cattle to graze it down to a lower level so it has a harder time regrowing. So maybe once you apply this just for one instance, maybe 3-4 years, you can go without it. And then any escapees from the grazing, not from the Roundup, but any escapees from the grazing that are able to reach maturity, they will begin to enlarge in size and the animals won't graze it any longer. But the younger ones, they can be knocked back by grazing. So one application could be enough for cleaning up the pasture in the short term, and another application might be needed 4 to 5 years later on reestablishment. So we don't know how high the reestablishment rate would be because we haven't looked at it yet. But if you clean it up once, then as the density increases in future, and that will be 4 to 5 years later, you might have to retreat them. So this strategy can save a lot of time and money by avoiding reseeding, mowing, burning, while also improving the forage productivity. So this is a clump of wireweed about 8 months later. You can see maybe one or few here green stems, but the rest of the clump is dead. And you can see clovers and other grasses, Bermuda, annual bluegrass trying to take over and grow in that area. So this is a picture from the actual application on the 10th of July in 2020. And these are those 50% treated plots. You can see this line here from here to here. So that's the plot that had the 50% single pass. And you can see these dead and weathering or weathered clumps of wireweed. So they are no longer alive and they are just disintegrating and they will fall over. The grasses will take over. This is the neighboring plot that had 25%, and you can see a little bit more green in there. But the strands that did die, they are also weathering. It's only the ones that didn't die, they remain green. So forage regrowth and dead wireweed about a year after treatment. So this is the following summer. So that where an entire clump used to be, all those strands are dead, and they are lying down on the ground and decomposed. Then you have animals, cattle moving, excreting. So all those microbes, they will begin to decompose these high lignin and high cellulose or hard remnants of the wireweed, and forages will take over. So I would like to thank the support from California State University, Fresno, UAL, who actually purchased the Wick Viper and made this study possible. Also the LSAM program for engaging the students, and the Department of Animal Science at Fresno State faculty and student workers who helped with the fieldwork. And with that, thank you very much for being here. And if you have any questions, feel free to share that in the chat.
Great, Ranjit. Well, I think first let's do the DPR poll.
Sure. So the question is, actually, they need to do the poll, and only then I can give the right answer.
Correct. So the first question, Roundup glyphosate is a selective herbicide, true or false?
So,
And then the second question, hold on, I think they need to answer first.
yeah.
Common rush is a perennial weed. True or false? And then submit. And then maybe we'll say the answers after a couple questions. So the first question, Ranjit, that came through is regarding common rush weed. Can goats graze this weed species? Is it attractive to them to graze?
Based on my experience, this is present in the sheep unit and the beef unit. Goats, they don't. Generally, goats are notorious for grazing almost anything, but for this species, not so much. Sheep, they do knock it down a little bit, but not a whole lot.
And then I just have a quick question. I have an incidence where I planted a non-crop area into a grass cover crop, and I had some breakthrough malva come through. And unfortunately, I had to, spray the whole thing with Roundup to really target the malva. So in that kind of situation, would this technology be effective if that was to be a crossover kind of thing?
So the thing with malva is that as it enlarges in size, and that's true for most weeds, they become harder and harder to control. And if you want to use this Wick Viper as a control strategy, then you will have to have that height differential. So if you have like a smaller area, then maybe a protected spray when with the cover on the nozzle that the herbicide doesn't bounce off might be better than waiting for it to grow larger. That makes it harder to control.
Mm-hmm.
And if everything else fails, like a weed whacker, any mechanical control.
Yeah, with malva though, yeah, comes back. Great, thank you. You can answer your questions now, so we know if.
So the first question is, the Roundup is a selective herbicide. The correct answer is false. It is a non-selective herbicide. It won't distinguish between a crop and a weed. The only option where it will act as a selective herbicide is where you have Roundup ready crops. So that's a transgenic trait, but in wild populations, it is a non-selective herbicide. And the second question is, common rush is a perennial weed, and the answer is true. Once established, it will stay alive. It will green up year after year and keeps on coming back in the same locations.
Okay, great. Well, I think that closes out our session, and we'll turn it back over to, I think, Florence. Thank you.
Thank you, guys.
No, I think we have a break, no? Yeah, we have a break from 9:20 until 9:35. So we'll meet you in about 10 minutes. Actually, it's already 9:26. So let's reconvene in about 10 minutes for session number 4, which is hot topics in pest management. So why don't you take a little break, stretch, and we'll see you in a little bit.
All right. Well, good morning, everyone. And we are at a little after 9:35, so why don't we get started? Welcome, everyone, to today's second session, Hot Topics in Pest Management. My name is Jake Wanger. I'm an assistant professor of entomology at Fresno State University and am one of the 2 chairs that helped plan this session. This year, I'm pretty excited that we are offering 2 pest management sessions at the Plant and Soil Conference rather than our usual one. And I really did enjoy the weed management session we just got through, some really fascinating research. And look forward to continuing our dive into the world of pest management. As far as this session is concerned, my co-chair, Maria Koivunen, and I decided rather than focusing on any specific area of pest management, that instead we were going to invite speakers that are investigating emerging issues in entomology, plant pathology, and weed management from across the state of California. I would like to take a moment to thank our speakers, Jhalendra Rial, Daniel Hasegawa, and Matthew Fatino, for taking the time to join us and to share their excellent research. As a brief reminder before we get started, this session is associated with 1.5 DPR continuing education units and 1.5 certified crop advisor units also. So we will be utilizing the polling feature at the end of each talk to collect answers. So if you are applying for those units with the DPR, please make sure that you complete those poll questions. If you have any questions during the presentation, please submit those to the question answer box found at the bottom of your Zoom screen. With that then, I will transition to our first speaker, who is Dr. Jhalendra Rial. Jhalendra Rial is an area-wide integrated pest management advisor with the UC Cooperative Extension and is an associate director for agricultural IPM for the statewide IPM program. Rial's current responsibility includes conducting applied pest management research and educating pest control advisors and growers about improved pest management practices. Rial's research focuses on multiple exotic and invasive pests of fruit trees and nut trees in California. He has received multiple awards, publishing over 20 scientific and 100 extension articles, and has given over 120 talks in pest management topics. And with that, I will pass the presentation over to him and get to hear his excellent work with navel orangeworm.
Good morning. Thank you. Thank you. Let me share my slide here. Okay, well, great. Thank you so much. Thank you so much, Jacob and Maria, for inviting me for this talk. So I'm going to cover today about the work that I and my colleague Tapan Pathak and Mahesh Maske, they are climate scientists. We work together and also share a little bit of work that we're working not related to climate change, but related to navel orangeworm migration in collaboration with the Chuckbergs. Okay. Just a general introduction. I know that majority of the attendees probably already know about navel orangeworm in general. It's the major pest for across the all nut crops, mostly almonds, pistachio, and walnuts. And it's also these insect, the larval stages directly cause damage to the nut meat. They feed on the nut meat, so that would be the direct loss of the yield. And the indirect loss would be whenever there is a lot of infestation by the larva on the nut meat, that invites the Aspergillus fungus, which produce the aflatoxin, which is non-carcinogen. So it's not only just the direct damage, but also the indirectly contaminates the nuts that potentially with the aflatoxin. So because of that, it's a quite important pest. Just a little bit on background. So a navel orangeworm larva in the fall, late fall, the larval stage inside these nuts, they are still on the tree. And a lot of those nuts are still on the tree even after the harvest, right? So these are the leftover nuts in the tree. We call them mummy nuts. And these maturity stage of these larvae overwinter on those mummy nuts. And as you see on these pictures, so these are a lot of nuts that are still on the tree. That's where the larvae survive winter, overwintering. And so after in the spring, they pupate and the adult come out, and they spend that first generation and second generation, third generation. That's when they start infesting the nuts, fresh nuts in the orchard system. So just a little bit of overview of that, the phenology that I just mentioned. The first generation or the overwintering adults, this is a adult activity. They rely on not only surviving the winter in there, but also whenever they come out, the adult, they rely on these mummy nuts to go back and lay eggs there and spend that first generation completely on the mummy nuts. When the second generation, second flight, it starts, the second through fourth generation is the one that go to the fresh nuts when at that time the nuts are split. And that's where they go and lay eggs and cause direct damage. So again, the importance of these mummy nuts, not only the bearing the larvae in the wintertime, but also serving as as a substrate for egg laying for that, for whole first generation survival. Just a sort of overview of the how we manage the navel orangeworm overall, because it's a very difficult pest to manage. So the cultural practices like removing those mummy nuts, basically reducing that pressure going into the spring, would be the foundation of navel orangeworm management. Of course, we use the various kinds of traps and monitoring tools in the spring. We use the mating disruption if that's as a part of the program. The mating disruption for navel orangeworm will be included in the early part of the season that covers throughout the season. And whenever, if there is walnuts, there are a lot of other factors also in the first season when there are curling moth damaged nuts, sunburn, blighted nuts. If those nuts are there, navel orangeworm take advantage of that, even those walnuts at that time is not splitted. So that would be another factor that minimize those damage, would also help to minimize navel orangeworm damage. If you go to the second through fourth flight, and in almonds, if you think about the 1 or 2 insecticide application, it's common because that's when the nut splits and navel orangeworm comes in and do a direct damage at that time. And in walnuts, this is not a major concern because they are not ready yet, or they are not splitted yet for this flight. But if you go to third and fourth flight, that's when the walnut become vulnerable, and same thing with the pistachio. And that's when there is need, then insecticide goes in. So if you think about this all together, even though we're following these practices, growers are following these practices, it's not enough. And it's very unpredictable pest. Some years it just, the damage becomes so high statewide. And with these, 2.5 million acres of the crop, it is very difficult to navigate. So as I mentioned, it's a very opportunistic pest, multiple generation, currently 3 to 4 generation, well adapted to all different kinds of environment. As you see, they survive on even that poor quality host, which is mummy nuts, right? And then they can switch their host from one to another if they get opportunity. They are highly mobile, migratory from one place to another place, depending upon the need, depending upon what's available out there. High reproductive potential and highly temperature dependent. So that's why navel orangeworm is such a difficult pest to manage. One of the survival strategy I kind of think about navel orangeworm is it's more of, it's a little unique compared to something like similar another moth or the larval pest that we deal with in walnuts, especially curling moth, right? So navel orangeworm lay eggs 300 or plus eggs in some cases, versus the curling moth, it just lay eggs 30 to 60 during that time. And when you looked at navel orangeworm eggs, they dump their eggs anywhere, whatever the feasible, even mummy nuts and other places, versus the curling moth is very careful where they put their eggs. So their survival strategy is different. Even though a lot of these larvae, when they're hatching out and making into the adult, is about 10% in navel orangeworm, still, because it's a number game, right? It's still their number is exceeded compared to other pests. So that's why it's critically important to put into the perspective in terms of the role of the various factors in building their population. We also recently worked a little bit about their migration behavior, especially the walnut versus almonds scenario. And essentially, we're looking at the fatty acid content of the almond versus walnuts, that's the signature difference between these two. And that difference is translated into the moth, the fatty acid content in the moth when they grew up as whether they grew up as a larva in the walnuts or almonds. So in other words, when you looked at this moth and looked at the fatty acid profile, you'll be able to differentiate between whether they fed on almonds or walnuts in short. We looked at these different scenario, almond dominate landscape and walnut dominate landscape, and we put the traps in the walnut orchard, but in these two different scenarios. Walnut orchard in walnut dominated, almond orchard in almond dominated. The walnut orchard in both cases, but whether it's almond dominated area or walnut dominated area. So these are the different regions that we cover: northern part of the valley, Sacramento Valley, and southern part of the valley, where you can see the walnut dominated areas and then also the almond dominated areas. We put the traps of different kinds here. You can see the OB bait trap, which is a Peterson trap. It's a female attractant trap. It has a pistachio bait in it. It's a food-based attractant, phenylpropionate, and then also the pheromone. So we put these 3 trap type. In the orchard scenario, we put 6 of these OB bait trap, and middle part is the pheromone trap, and about 50-meter distance we put the PPO lure. And so we had 6 orchard, 3 with almond dominated and 3 in the almond dominated landscape in 3 regions. So total 18 orchard, and we did this work for 3 years. So what we found was when we looked at these moths, dissecting them for the fatty acid content, we see that landscape had the impact on male migration. Mostly that's what we're seeing. In other words, the pheromone trap capture, when we looked at it, we can see in here that these are on the graph, you're seeing the basically percentage of navel worm that is developed in walnuts. But the red color one is in the almond dominated scenario, and the blue one is walnut dominated scenario. So in that case, when you looked at the first one, pheromone ones, right? So if it's almond dominated area, you are catching less percentage of the moth that is coming from walnuts on that resident orchard, compared to when you looked at just the walnut orchard, and it's dominated by the walnuts. Similar, but we did not see that kind of discrepancy too much on the OB bait, which is local more. It has a less active space in terms of it's volatile, and so maybe that's the reason we did not see much difference. So in other words, we're seeing more or less about the same percentage of the moth that we captured in walnuts, regardless of the landscape type. And we think that it is coming from a majority of them coming from the walnuts. PPO lure is a little reverse. So maybe if I could put the next one. So in general, if we put all combined together in these 3 years, 19, 20, and 2021, we see about anywhere from 31 to 60% of the moth that we captured in this walnut orchard, actually there are larvae developed in walnut. So in other words, about altogether, it's about 45% of the moth trapped in walnuts were developed as a larva in walnut. So the rest of the percentage coming from somewhere else, right? But it's still, I mean, these are the resident population of the walnuts, of the navel worm in walnuts. So it makes sense that when we have these sort of 2.5 million acres now, the total acres of the nut crops in the valley, and we have walnuts and almonds and pistachio all together, and it's very likely, or very sort of like a general way of also we're thinking that they are navel worms in host of all of this, and they can migrate from one host to another host. And we kind of more or less saw the similar thing here too. So in this case, we're looking just walnut versus almond. But of course, if you have the walnut towards the middle of the almonds, you expect a little more abundance of the population in your orchard. But it doesn't mean that 100% of your population is coming from somewhere else. So it has some influence on the population based on what's out there. Okay, so now moving on to a little bit about the work, that climate change work that we did. I'm not talking about definition of the climate change here, but I think that in a climate change, when we talk about the 3 parameters is important: precipitation, carbon dioxide level increase, temperature increase. And precipitation, in a sense that it's more of unpredictable. Sometimes it's too much, sometimes too little, sometimes too much. So, and also all of these factors had some role in terms of insect activities, right? So for example, if you think about the drought and lack of precipitation, And we have those a lot in California, so it's nothing new for us. In some cases, they're favoring the development and growth of the herbivore. In other cases, they can make the plants attractive to certain insect pests, but it's not always the case. So there is a little bit more complex interaction, and we need to look at individual species differently for this aspect, because not everything is kind of equal or similar. In terms of the carbon dioxide, the role that I found based on my the literature search and other things is that increased carbon dioxide level can influence the insect fecundity, insect consumption rate, and other things. And also it can also increase the plant quality better, right? So if you make the plant quality better, that means that you likely have a more, you expect more damage or become more attractive to the herbivores too. So again, we need to go these things in a little bit more this is just a general statement, right? So may not apply to every insect and every system. So if you looked at the overall, the influence of these temperature on the insect, of course, it increased the number of generation in many cases. We can see the expansion of the geographic range. That's where we're seeing, the insect that were not before in this region are more common now, and that kind of scenario. Other cases that you can see the outbreak of the diseases scenario. For example, last year we see a lot more curly top virus in tomato likely because of the bitleaf hopper population is triggered by the early drought and drying of all the grasses on the nearby foothills, and they just move to the valley quicker, right? So those kind of things could happen. Overwintering survival is another thing. The other interesting thing that I found was it's not just that, but also when there is prey and predator type of relationship, these temperature changes also alter or make them imbalance or sort of alter their relationship, and that also become a problem. So when we looked at the California conditions, then we know that we're dealing with these changes in temperature. One of the predictions says that California future statewide temperature will be increased by 2 to 7 degrees by the end of the century. And we all know the insect growth is directly related to the temperature. Here is one sort of like 2 slides I saw here, just the general temperature or expected temperature increase anywhere from 1895 to not expected, it's a historic 1895 through 2018, right? If you looked at the overall increase in the temperature on an average US, it's about 1 degree centigrade increase. But if you looked at the California, some of the counties, for example, Ventura County, the temperature increase was double of that, 2.6 degrees. Here I looked at the Stanislaus County because that's my home county in terms of my extension responsibility, and it's a 1.3 degree increase. So California were in that higher end compared to the U.S. average in terms of the past temperature increase over 100 years or so. So we looked at these all scenarios and together, and we found that navel orangeworm is, of course, the important pest, and we wanted to quantify the impact of the changing climate on navel orangeworm. That's why we team up with Tapan and Mahesh, and we did this work. Basically, on this model, we looked at the 23 California Central Valley counties. And the climate scientists, they routinely use these models, like the 10, something called the general circulation models. That's where we use these models as a sort of like to get the temperature, future temperature, and then use that in the model. Again, in each county, we have 5 different representative points that where we took these temperature data, and then we also use the 2 emission scenario. That means that the projection when you think that the climate change will happen, but the people will have aggressive mitigation measures in place. In that case, it's called the 4.5, the RCP. And if we think that in the future, the climate change will happen, but people will not take that much of a step to mitigate it, that would be even a more aggressive or intensive scenario, which is 8.5. So of course, for the insect side, we used all the information that we know. It's already been published in many different literatures, many different scientists already worked on it. So we use these bases that is already we know about, number of generation, when they start their activity. All those information were kind of already present, and we use those information. For example, one of the things that we know is for the first and second generation of second and third generation of Navel orangeworm, when they grew up in these fresh nuts, in almonds and walnuts, it takes about 444 degree days for them, versus when they grew up in pistachio, that would be less than that. So we use these two separate things for modeling pistachio separately and almonds and walnuts together. So we use these all different methods, the steps that I'm not going to talk too much about, but in general, so we have these models, degree day models. We try to relate that to the future temperature and then try to see the number of this generation and the influence of those future temperature to Navel orangeworm population. One of the things that we looked at is, of course, the number of generation in almonds and walnuts. So you can see in this case here, the under 4.5 RCP, which is where we will implement some kind of mitigation measure, but still the temperature will increase in the future. So in that case, we can see at least, in some cases, we've seen that first generation of that, which is the adults that coming from overwintering in lakes and the mummies, for them, they will be able to complete their life cycle their life cycle for that first generation, the life cycle will be shortened by about 6 weeks in the future climate condition by the end of the century. For example, in Sacramento County, Navel orangeworm was expected to complete its first generation in almonds and walnuts is around 193rd day from January 1st, historical data, where in the future, they may be able to complete in, even in 154 days to 180 days from January 1st calculation. So that means, the duration for completion of that first generation will be shortened, which is expected when you have the increased temperature. This is a pistachio, kind of more or less similar scenario. In pistachio, looking at just the example Tulare County, where we expect the 5th generations, and it's on historically, if you think about it, it's probably 292 day from the January. Now they can complete as early as 262nd day in this, based on this model. On an average, this is a sort of 3rd generation, 4th generation, and 5th generation. So on an average, the occurrence of the 5th generation Navel orangeworm, we found that at least under even 4.5 RCP, 3 counties in the southern part of the valley here, you can see they can complete their 5th generations as early as 2040, and that number is pretty much throughout, which moved to up to 17 counties by the end of the century. So the 5th generation Navel orangeworm will start appearing within next 22 years based on this model and the study. This is using the 8.5 RCP. In that case, even you're seeing more intense or more number of county, you see likelihood of 5th generation coming early, because this is a scenario where we assume that nobody will take any mitigation measure to address the climate change issue. This is for the pistachio. Pistachio even more intense because, as I mentioned, the pistachio it takes shorter time to develop the one generation, that second or third or fourth generation on pistachio compared to almonds and walnuts. So they need shorter time. That means that they can accomplish more number of the generation. And in that scenario, we have 7 counties that we may be able to find the 5th generation as early as 2040, and in 17 counties by the end of the century.
Just to let you know, we have about 2 more minutes, okay?
Okay, thank you. Yeah. So the increased pressure is more intensive in 8.5, as I mentioned. So this is a sort of like a scenario. I mean, it doesn't mean that, the whatever we predicted based on these models is going to be translated the same way. But at least there is enough evidence now that the future climate change will impact the insect population. One of them, important one, is Navel orangeworm that we know. The idea for using this one is just to provide a little bit background information, providing the information to the policymaker, to the industry, kind of think about how we can mitigate these measures, so how we can deal with these things in the future. Whether we need to think about investing more on the research on climate change and pest issue, or investing more resources on sort of like a futuristic plan for integrated pest management issues and things of that nature. So again, I mean, it's not the complete set, because we also did not look at the crop phenology tied into this model. That could be another factor can be looked at in the future. So yeah, as I mentioned, it's more of the sort of providing awareness about what is happening and how we can potentially think about or formulating the policy or plan in the future to addressing these issues, because 2.5 million acre plus, and by then in the future it will be expanding even more, and dealing with this one insect that is critically important. We need to think in advance. So that's kind of like notion of this publication and paper. Okay, well, looks like that's all I have. So I would be happy to take any questions. I can see the poll questions here. I'll answer them when they fill that poll question out.
Excellent. Well, thank you very much, Jhalendra. We will take a minute to fill out the poll, and as Jhalendra mentioned, you will answer the question. It looks, if anyone has any questions, feel free to submit them to the Q&A. Although it looks like we will need to transition. We've got about 1 minute, and there we're 10:05. So we'll be moving relatively quickly into Dr. Hasegawa's talk.
Well, thank you so much. And I would say that thank you so much for inviting me. I have another talk in about 25 minutes from now, but it's in the same place, so I'll be okay in that. But I will not be able to answer those questions, but I'll be happy to, whenever you get the questions, if you send it to me, or I will be happy to do that. So
excellent, thank you.
Thanks again. I really enjoyed this meeting. This is my second time I'm presenting in this meeting, and I really like this group. It's a sort of internal cohort. It's close group, and I always enjoy to be here.
Okay, Jhalendra, before you go, I know you're busy to present in your next appointment, but can you go over the correct answers for the DPR questions?
Yeah, definitely. So it's a challenging pest to manage, because that's a right answer. All of the above, right? So there are many multiple factors playing into role. For the first one, and 92% got right. The second one is, yeah, that's right. As early as 2040, we might be able to see the 5th generation of Navel orangeworm completion in the southern part of the valley. That's true. So thank you.
Very good. Thank you so much. Okay, Jacob, you want me to jump in here?
Yes, please do.
First of all, introduce myself. My name is Marja Koivunen, and I work in the biopesticide industry as technical manager, product development. and I'm the co-organizer for this pest management session. And I would like to introduce our next speaker, who is Dr. Daniel Hasegawa, and he comes to us from USDA in Salinas, where he is working as a research entomologist, and his training and education is in biochemistry and molecular biology, and he has recently engaged in the research around thrips control, and to develop new tools and manage insect pests and insect-borne viruses that are affecting vegetable and fruit production. And as I said, or maybe I didn't say, but we picked this topic because we know that Salinas Valley lettuce growers have had really bad problems recently with the with thrips, and of course the pathogen that they are spreading. So welcome, Daniel, and take it away. Thank you.
All right, great. Thank you. Can you hear me and see my screen okay, Maria?
Yep.
Thank you so much. Thank you, everyone. And thank you to the organizers and the chairs, Jacob and Maria, for this wonderful opportunity. And so really the purpose of my talk today is to provide a broad overview of the challenges and the problems to managing thrips and Impatiens necrotic spot virus, which is a virus that has been around since 2006, but only until recent years have we seen it reach really severe economic impact levels and severity and incidence throughout the valley. So I'll touch a little bit about the problem, the challenges, and then dig a little bit into the research that we're doing and the plans that we have going forward. So I started here in 2019, and so I don't have a strong historical background in this system, but my experience with this issue has really started since 2019, and it's been a really big focus of my research program so far. So, and I think anyone that's familiar with vegetable production and leafy greens production has a good sense of how important it is within our county of Monterey County and throughout the Salinas Valley. It's one of our major commodities, has some of the highest acreage within the valley. But in addition to lettuce, there's all these different crops, over 100 different crops that are grown, which from an IPM perspective, you can imagine can be challenging to develop and improve some of these management approaches, especially when you're dealing with a pest and pathogen that has quite a large host range. And so these are some of the foundational challenges that are presented just for managing thrips in general. And so I'll talk a little bit about how that applies to the culture of lettuce production here in the Salinas Valley. So the production season is quite large. It's quite long. It runs, planting typically starts in January and runs all the way to November when some of the last harvests are occurring. In 2020, I think 20% of all lettuce was produced organically. And so immediately there are restrictions in terms of how we think about IPM and managing both the pest and the pathogen based on the requirements for organic versus the other options that are available for conventional management. So because of the long season, because of the relatively short crop cycle for lettuce, you can see there's actually 3 different plantings pictured in on the screen right here in the back. We're going through a harvest period. We have a crop that's about midway through its crop cycle, and then in the foreground here we have lettuce that was direct seeded probably a week, 2 weeks ago, and it's about to undergo the thinning stage within the next week or so. And so throughout the lettuce season, a grower can get about 2 different plantings, consistent rotations with cole crops, as well as a number of other crops that can be integrated into those rotational strategies. And so the intensity and the dynamic nature of the entire cropping system, not only within lettuce, but integrating other crops is quite complex. So brief history of this virus. So this picture was sent to me from Gina Kulfer, who is one of our PCAs here, and she's been a great collaborator on some of these projects and helping me understand some of these issues here. And so this virus was actually first described in the Netherlands in 1980. And then in 2006, as I mentioned before, it was first reported in Monterey County in lettuce. Historically, it's been an issue in ornamental crops. Within the last couple of years, it's really emerged and has been recognized as a major pathogen that's affecting vegetable production in North America as well as Europe. Minor to severe isolated outbreaks since it was first documented in the county. And then, as I mentioned, in recent years, severity has increased, amounting to up to 100% crop losses, millions of dollars lost. And then last season, we actually first identified this virus in another major lettuce producing region, which is Arizona and the southern desert regions of California. And between this area and that region, I think it accounts for about 90% of all of US lettuce production. So quite a large contribution. And so we continue to monitor the situation and the emergence that's happening in Arizona. And that's working with John Palumbo and Stephanie Slinsky and Bindu and their wonderful team of extension personnel down in the desert. So I'm going to show you some different pictures of the virus and different looks of it. This is in romaine lettuce. So they call it Impatiens necrotic spot virus because of the severe necrosis that you see on the leaves of the plant. It also causes stunting of the plant as well. And so you can see this plant right here and this plant right here is much smaller than this one here. There's a latent period of about 7 to 14 days before the virus actually starts showing symptoms within the plant. So I can't say definitively that this plant is not infected, but at least it appears to be healthy compared to all of these other plants that have been infected by this virus. This is a field taken in 2020. And you can see quite a bit of damage caused by this virus. This was about, I want to say 7 weeks into the crop production cycle. During this planting slot, it was this field went for about 9 weeks. Quite a bit of damage that was suffered within this field. In 2021, this was considered a total crop loss. You see some of these plants are extremely small, meaning they were infected at quite a young age. They were stunted in their growth, and you see different stages of infection throughout this entire field. And this was about a 5-acre field. In 2019, when we really first tried to document some of the severity and the epidemiology of this pathogen, we were turned on to this field, which led to several other studies. But what I really just wanted to show here was how early that this pathogen can infect lettuce. And so this is at the pre-thinning stage, a couple weeks into the crop cycle, and you're already seeing very severe necrosis on the leaves. We did some monitoring studies as well as some cost analysis and economics that were tied to some of the losses induced by INSV. And within this, I think this is about a 9-acre field. And so total crop losses, both from not only the gross production losses, but also the investment that went into operational costs, the total losses amounting to about $110,000 just for this one field. In 2021, the Grower Shipper Association of Central California launched a task force, which was addressing one of 2 major pathogens that have recently devastated the industry, and that was Impatiens necrotic spot virus. The other one was Pythium wilt. I'm not talking about that, but we have another great collaborator, JP at CSUMB, who's working on that issue, but I will not be talking about that today. So for INSV, we developed an industry, it was a voluntary survey that was launched. And so we had over 70 members participate at our first meeting, and that involved researchers, extensionists, stakeholders, as well as county officials in Monterey County. And we had reporting throughout the entire 2021 season. And really the final numbers that we saw is that over 750 fields reported incidence of this virus greater than 1%. The thing here is that only about 25% of the industry reported, and so these numbers are actually quite underestimated in terms of the widespread incidence of this pathogen. When we look further into one grower, we did some additional analysis because of the thoroughness and the details that they provided, but of course, I'm not going to disclose any of the private information there, but the one grower reported 23 fields, which represented about 220 acres, ranging from 3 to 100% incidence, with an average field incidence of 30%. As I mentioned, this pathogen is spreading into other lettuce producing regions. And during the 2020 season, we estimated that pest management costs increased 10 to 15%, and that was primarily associated with pesticide applications, both for INSV, but also for the second pathogen, Pythium wilt. So Chris Valadez and Mary Ziske, both with Grower Shipper Association, have been really critical to this documentation here, as well as all of the task force members that have been a part of this really integrated team that we have. So one thing that I will mention now is that all of this pathogen is obligately transmitted by the Western flower thrips. It is the vector for the virus. And so we're dealing with a very small insect. You can see this young lettuce plant is infested with Western flower thrips at a very young age. And you can see damage, you can see the actual adults that are feeding on the plant as well. And we know that transmission of the virus can occur within several minutes of feeding. And so if you think about a management strategy, it becomes very difficult not only to manage the vector, but also the virus. So we'll talk a little bit about the life cycle here. I won't spend a lot of time into it. For anyone that's worked in the Central Valley or even other crops outside of tomatoes and lettuce, you're quite aware of the problems that thrips can present here. So really the important thing is that the host range for the Western flower thrips is in the hundreds. And so very similar to the previous presentation, this idea of having a wide host range can present management challenges for one particular pest. The insect goes through several life stages. The important part here is really its role as a vector for tospovirus. So tomato spotted wilt virus, which is quite prevalent in the Central Valley, and then Impatiens necrotic spot virus, which we find the majority of the time, probably 99% of the time, in lettuce. The other 1% is tomato spotted wilt virus. So really it's INSV that is our major pathogen here, our major tospovirus here. For those of you that are going to get DPR credits, I would pay attention to this important part right here, because it may or may not show up in your questions. But what's unique about this vector is that the virus must be acquired as larvae. So it has to feed on an INSV infected plant at the first instar, as well as the second instar. But in order for transmission to occur, they must acquire the virus as a first instar, complete their life cycle, and it's only those emerging adults that can transmit the virus. So only the adults can transmit the virus, and the virus is not transmitted vertically down into the egg. And so they have to reacquire it every single generation. So the other important thing here is that the host range for INSV has also been described in the hundreds. And so there's a lot of different cultivated as well as uncultivated plants that can contribute to the host range and the ability for this insect to acquire the virus. Let's see. All right, so I'm going to just kind of fly through the rest of this for the sake of time. So we run a Salinas Valley monitoring program, and so we have 3 years of historical data here. And what I've done is just plotted it on this graph with relation to the lettuce season. And if you think about production and temperatures as we progress through the season, that study that I demonstrated in 2019 was within this planting slot, right as thrips populations continue to increase throughout the summertime. So They're quite poor flyers, and so we've done a little bit of studies looking at dispersal patterns. We're still working on some other aspects of this, but the bottom line is that we've been able to capture thrips pretty high up into the airstream, and so up to 25 to 27 feet. If anyone's been here in the summertime in the afternoon, you know that wind speeds can get up to quite high speeds, especially as you move further south through the valley. And so being wind dispersal, dependent on wind dispersal, we do think that the wind is playing quite a large role in terms of the abundance and dispersal patterns for this vector. I just talked about this beginning part. I'm going to talk briefly about the epidemiology and host range, and then these are just going to stay as bullet points at the bottom. And so my talk will be done fairly shortly. So field surveys, I mentioned this was the production cycle, and so we've conducted several different surveys during different periods to really address the host range for INSV during not only the lettuce season, but also during the off season. And so the important point here is this last period, we conducted field sampling during what is called the lettuce-free period, where the county established years ago to really mitigate another lettuce virus, lettuce mosaic virus, and that's served to be very helpful in terms of managing that pathogen. But we wanted to address whether these alternative crops are actually playing a role in bridging the virus when lettuce is no longer in the valley. So sampling, we did this by triple antibody sandwich ELISA, so a serological test, and then we also did some validation using PCR from a lot of these different samples. And so we have different sampling sites. The important part is the East was kind of our hot zone in 2019. The southern regions, we didn't see as much occurring down there. And so we wanted to focus sampling in some of these different areas. We've sampled over 3,000 samples, 73 species. We've identified a top 10 host based on percentages of infection. Again, for some of you that are doing DPR credits, this is an important slide for you. So these different plant hosts, little mallow, sow thistle, lamb's quarter, shepherd's purse, all of these are demonstrated hosts for the virus. When we look at those top 10 lists and looking at their percentages of infection during these different sampling periods, we have pretty strong evidence that the samples that we are collecting specifically during the off season are serving as reservoirs for the virus. And so we're working with the county as well as the entire industry, as well as outside of the lettuce commodity as well, because weed management is going to have to be a systems-wide approach as we move forward. So just some additional projects, not going to spend much time here, but working with UC Davis and Riverside and a lot of other organizations and academics across the country to try to develop some solutions. And that ranges from augmentative biocontrol to cultural strategies, as well as integrating some genetics and biochemistry and biotechnologies for the future. And so with that, I think that's all I have. Just about 5 minutes before the end of my allotted time. I just want to thank my technician, my students, all of the great collaborators, as well as different various funding sources that have contributed to this work. And finally, the growers and PCAs who have been absolutely critical for us to really start understanding the epidemiology of this issue. So Jacob and Maria, that's all I have. Thank you. Would you like me to read the questions for the DPR pool?
I think we have a little bit of extra time. So yeah, why don't you do that? Thank you.
Okay, so the first question is, which of the following is true? Thrips larvae cannot transmit the virus. Only adult thrips can transmit the virus, being INSV. INSV must be acquired as larvae in order to transmit the virus as adults, or D, all of the above. And then the second question is, which of the following weeds are described hosts for INSV? Little mallow, also referred to as malva or cheeseweed, annual sow thistle, lamb's quarter, shepherd's purse, nettle leaf goosefoot, or all of the above.
Okay, and we will let people answer those questions. And we can continue our chat here. There is one question in the, I saw in the question and answer section, and it is a question about why can't the larvae, the instars, the small larvae, why can't they spread the virus? Why is it only the adults?
Yeah, so that's a great question, and there's still the exact mechanisms for that is still being worked out, not by my group, but a lot of other groups, Diane Ullman at UC Davis, and then Anna Whitfield and Dorth at NC State. They've really worked on some of these different aspects of virus transmission and the mechanisms of it. But basically what happens is when the larvae acquires the virus, it only localizes into the guts. But for transmission to occur, it actually has to make it back into the salivary glands. And so it's during those different life stages that the insect has to go through. And it's only during those times when the virus is able to make it back into the salivary glands. So same thing with the adult. If the adult feeds on an infected plant, it ingests it, but it never makes it back into the salivary glands.
Oh, okay.
Yeah.
That makes sense. Okay. I didn't see any other questions in the question and answers section, but I have a question.
Sure.
So it is obvious that those all kinds of weeds around the field, they can serve as the reservoir for the virus. But does the plant have to be actively growing to actually host the virus? Meaning that if you have lettuce plant residue that got infected during the growing season and it's left in the field, can that be a source for virus as well?
As long as, so the short answer is yes, it can, but under the conditions that the plant is healthy enough in the sense that the larvae are still able to feed on it. So if it's a completely dead plant, now you're really talking about, where is the female going to lay her eggs? Because that's only under those conditions are the larvae going to emerge and then feed on the infected plant. Yeah. Yeah.
So does the virus, does it actually survive in the soil without a host?
No, it does not.
Okay.
Yeah.
All right.
Yeah.
So I think we have the poll.
Yeah. So question one, which
Yep.
of the following is true? And actually all of them are true. So the larvae cannot transmit the virus. Only adults can transmit the virus, and it must be acquired as a larvae in order to transmit as an adult. Yeah.
Okay. Very good. And the second question?
The second one is all of the above. Yeah. They're all described hosts for INSV.
Okay.
And one thing I'll say, and I don't want it to be misleading, but where we do our samplings and we see those weed reservoirs, those in general, they're not issues for lettuce growers. It's really in these areas that they don't have access to manage. And so the industry has done a great job with weed management, but it's going to require education and broader applications beyond the control of the lettuce industry.
Right. Yeah, that makes sense.
Yeah. Thank you.
Thank you. Thank you, Daniel. This was a good presentation. I learned a lot. So thank you.
Thanks. I appreciate it.
Okay, thanks. And we are ready to move on. And our next speaker is going to be Matt Fatino, and he's a grad student at Brad Hansen's group at UC Davis and the Weed Science group. And he started his graduate studies back in 2019, and he hit the jackpot, I would say. He started working with a weed that has become really problematic lately, especially here in the Sacramento Valley. Branch broomrape in processing tomato. So Matt is currently studying for his PhD, and he's continuing on the same subject. I know he has been a very sought for a speaker lately. I think he presented in the Wheat Science Conference as well. And this, I would consider this topic as one of those what's old is new again type of topics that we had yesterday with Janine Jones. Matt will tell us more about this weed, but we thought it was already eradicated, and it has showed up again within the past couple of years in the growers' fields, and we are running out of time to get some solutions. So Matt, if you're ready, please.
Yes, thank you for that introduction. Can you see my screen?
Yes.
Okay. Hello, everyone. My name is Matt. Again, I'm with the Wheat Science Group at UC Davis, and I'm going to be talking about some chemical control strategies for branch broomrape in California processing tomatoes. Here is just an overview of what I'm going to talk about today. I'm going to introduce broomrape and its background and significance. I'm going to briefly talk about the California tomato industry. I'm going to introduce the Pickett decision support system, and then I'm going to talk about some crop safety, rotational, and efficacy studies that we did in 2019, 2020, and 2021. And then I'll end with some future work. Branch broomrape is Phylloponchia ramosa or Orobanchia ramosa, depending on which taxonomic camp you're in. It is native to the Mediterranean countries. It's an obligate root parasite. It utilizes a hostorium to parasitize host plants' roots, and it has a wide range of agricultural host species. It can infect brassicas, the solanaceous crops, Fabaceae crops, cucurbits, and asters. And the kind of big reason why it's such a big deal is that it's a regulatory nightmare, and it's an A-listed noxious weed in California, which means that the field, if reported, has to hold orders placed on it, and it's quarantined and crop destruct generally. And then you can't grow host crops in that field. So here's a kind of a schematic of broomrape's life cycle. I really want to focus on this below ground, as this is 2/3 of its life cycle, kind of why broomrape is so problematic. Starts out as a seed, obviously, and they're very, very small, like 0.2 millimeters or less, kind of the size of like ground pepper or espresso grounds. They receive a stimulant from host roots. In most cases, that's strigolactone. They germinate and they attach to a host root, and they form a tubercle, just like a swollen nodule on the host root, and then they emerge. And from emergence to flowering is very quick, can be a couple weeks. And then once they flower, they set seed very quickly, and they can produce a couple 100,000 seeds per plant. So very prolific. Here's some photos of broomrape in the field. This was from our test plot in Yolo County. Here is broomrape attached to a volunteer tomato plant. We also saw it this past season on nightshade. So you don't even have to have a host crop in the field. You can have a weed in the field, and it'll attach to that. Here is it on a little bit like a transplant. Here is it on a more mature tomato plant, and this is just a picture of it from the field. It's pretty small statured. I never saw anything over 12 inches for the most part, and it's really hard to see. If you're not looking for it, you're probably not going to see it. So you definitely can't see it driving by your field. You have to go looking for it. There's a huge risk of spread in California. California shares a similar Mediterranean climate. We grow a diversity of hosts, as you all know: cabbage, carrot, sunflower, potato, tomato. We have pretty much no control options as of now, and it has super high seed longevity, 20 to 30 years in the soil. Huge. The tomato industry specifically is very, there's a lot of custom operations, and it's very consolidated. So we have machinery moving kind of all over the state and in tens to hundreds of fields per year. So that's kind of problematic as far as dispersal of the very small dust-sized seeds. And once again, 2/3 of its life cycle is below ground. So you don't know you have an infestation until it's too late. Okay, California tomatoes. We produced over 90% of tomato production in 2020. That was 11.4 million tons across 230,000 acres. So it's a big industry. Tomatoes are transplanted, they're drip irrigated and mechanically harvested. And the two main growing regions are the San Joaquin and Sacramento Valley. Top 5 counties are Fresno, Yolo, Kings, Merced, and San Joaquin. As was previously stated, Yolo County in the Sacramento Valley, but Yolo County has had numerous reports in the last 4 to 5 years, including last year, of broomrape. So it's a problem for sure, and it's getting to be more widespread. The picket decision support system was developed over 20 years of research in Israel to manage Egyptian broomrape. So it's sister species in their processing tomatoes. It's based off of growing degree days, and the treatment regimes reflect different infestation levels, but most involve a pre-plant incorporated application of sulfurylsulfuron, followed by multiple chemigation applications of imazapic. And none of the herbicides used in that program are registered for tomatoes in California. So picket control, just to give you an idea, I think they had a study in 2018 in over 30 fields, and they had up to 95% control of Egyptian broomrape. So it's pretty good. This yellow box is an untreated plot. You can see all the broomrape coming up. This is a very, very heavily infested field. So it works well in their systems. The image on the right is a little bit intimidating and busy, but that's just to give you an idea of the decision support aspect of this program. It's like a flow chart. I'm sure you've all seen those. In 2019 and 2020, we evaluated 3 picket programs. One was for a mild infestation, which was 2 foliar applications of imazapic. And then another was for moderate that involved a PPI sulfurylsulfuron application, followed by 2 imazapic chemigated applications. And the final one was intense. That was PPI sulfurylsulfuron and 5 chemigated imazapic applications. In 2021, we shifted our focus to imazamox and really only evaluated the intense infestation programs. I'll give you kind of our idea behind that in a little bit. In 2019 and 2020, we conducted 3 crop safety studies in Davis on campus in uninfested fields. So this was just testing the crop safety of the picket program. We had 8 treatments that were imazapic focused. We planted Heinz 1662 in a single plant line. In 2019, we had 2 studies, one planted in the beginning of May, one at the end of May. And then in 2020, we had a study planted in mid-April. So this is kind of busy, but I put some colors here to try to make it easier to comprehend. The red is the intense regime, the yellow is the moderate regime, and the green is the mild regime. And we had 1x of each of those, followed by 2x rates of each of those. And that's, again, to test the crop safety of this. And I'll walk you through one of these here. This treatment number 3, sulfurylsulfuron, PPI, again, pre-plant incorporated, and it's applied right before transplant, then mechanically incorporated. Then we have imazapic, chemigation, times 5 times at 4, 5, 6, 7, 800 growing degree days. So here we have the yield, tomato yield from all 3 of those studies across 2 years. There's not really a whole lot to see here, which is good news in that there were no differences in any of the treatments in any of the studies. So this supports the Israeli data that imazapic and sulfurylsulfuron are fairly safe on tomatoes. In 2020, we conducted an infested field efficacy study, and this was in Yolo County. This field was reported to be infested with branch broomrape in 2019. So we were lucky enough to be able to get in there and conduct a trial. We planted BQ271 in 2 plant lines and 12 treatments that were imazapic focused. Okay, similar idea here with the colors in the treatment list. We had 1x and 2x of those imazapic picket treatments, as well as an intense regime with 3 different imidazolinones. So we had imazamox instead of imazapic, imazapyr, and imazethapyr. These were kind of used to hedge our bets in case imazapic wouldn't be able to be registered in the state. And we also had foliar rim sulfuron. So in late May, we saw our first broomrape emergence. Gene Meow is here, and here am I, and we're looking for broomrape. As I said, it's pretty difficult to see. You really have to dig into the plant canopy to see it. We'd mark each individual cluster with a wire construction flag, and we used colors to designate weeks to kind of track emergence over time. So in June here, you can kind of see there's a few more colors out there in the field. In July, there's even more colors and more flags. And then here we go. This is in August. So this is right before we terminated the trial. There's thousands of flags out there with thousands of broomrapes. We had over 2,700 broomrapes in just 2 acres. I'd say this is probably a moderate infestation as far as infestations in the Mediterranean go. So it could be worse, but it's definitely enough, and bad for a tomato grower, good for a broomrape researcher. Here's the data from that study. On the y-axis, we have the number of broomrape clusters per 30-meter plot. Plots were 100 feet long. Not a whole lot to see here. We did get lower broomrape in picket treatments. So here's a sulfosylfuron plus imazapic, and then sulfosylfuron plus various imidazolinones. Those are what I'll call picket treatments for today. The non-picket treatments had more broomrape, but we couldn't find any differences among the treatments. The broomrape emergence in the study was pretty spotty. It was blocked, but still it was like treatment 8 had great control, but it shouldn't have had very good control. So more studies needed to be done to kind of parse about differences among treatments. In 2019, we initiated a rotational crop study in Davis in an uninfested field, and this was to evaluate the residual herbicide effects on commonly rotated crops. So a tomato crop received 9 picket treatments in 2019, and then we planted 6 different crops back in 2020. So we planted wheat, melons, beans, corn, sunflower, and safflower. Treatment list here, we have sulfosylfuron at 3 rates, imazapic at 2 rates, and then those 3 imidazolinones at a 2x rate, kind of to simulate worst case scenario. This is busy, but I just want to focus your attention on these red arrows to these green bars. That's corn. Corn was injured by sulfosylfuron at the 1x and 2x rates. That makes sense. It coincides with the label for sulfosylfuron. So if that becomes a treatment that's used for broomrape, then growers are going to have to figure that into their rotational crop and their rotations. There was also some differences in melon biomass, but that can't really be attributed to the picket herbicides. We had really bad gopher pressure as well as field bindweed. And we also put a rim sulfuron application down before we planted, which isn't registered on melons. The results, I kind of already went through those. This image on the left, I kind of to hammer home the injury on corn. So corn was stunted and had pretty bad chlorosis. So this is like knee to waist high corn. That was a sulfosylfuron treatment, where in the background you can see there's overhead high corn. So it will damage corn, sulfosylfuron will. In 2021, we pivoted to imazamox. Imazapic was kind of a dead end as far as registration in California goes. And this is in the same field in UC Davis. It's not infested. And this is, again, just crop safety. We had 10 treatments, again, imazamox focused. We planted 9024 SVTM in a single plant line, and we had 1 planting in late April and 1 in mid-May. We focused on only the intense regimes, kind of trying to throw everything we have at this. So we figured growers would want information on the intense regimes. We had 1 through 4x rates of imazamox because we knew that imazamox has greater injury on tomatoes than imazapic, so we wanted to be able to quantify that. And then we had 1 alternate timing to kind of see if there's any difference in broomrape control. These results here are tomato yield. They're a little bit misleading. There were no differences in either treatment among treatments, statistical differences. However, I can tell you from being out there in the field that imazamox at the 2, 3, and 4x rates will damage your tomatoes. There's no doubt about that. And you'll see an image in the future in a little bit that shows that. The injury kind of presented itself as stunted plants, like pale green stems, as well as just like really bad leaf curling. And yes, imazamox will damage your plants. That's kind of what I want to hammer home here. In 2021, we had another infested field efficacy study in that same field in Yolo County. 12 treatments, imazamox focused, 9024 SVTM was planted in one plant line, and it was planted really, really late in June. We were probably the last field in Sacramento Valley to be planted, and that was followed by probably 10 days of over 100 degrees. So in this image here on the left, you can see some very sad tomato transplants. Here we had 1 through 4x of imazamox in the intent regimes, as well as 2 alternate timings for imazamox. We included 2 legacy imazapyc treatments. One was an alternate timing as well. And then we had rimsulfuron injected through the drip 3 times, as well as Actigard, which is acebenzolaris methyl. That is a plant defense activator. And that's been shown to induce resistance to broomrape in like sunflower and tobacco, but not yet in tomato. So we wanted to see what that did. Here's our yield from that efficacy study. And here is kind of my point as to the damage from imazamox. There's, again, huge crop injury here. In the 3x and 4x of imazamox, even 2x, we saw injury. Significantly decreased yields and just very sick looking plants in general. I'm kind of disappointed to report that we did not have any broomrape in this field. There were 2 studies in that field. One at the south acre was the chemigation study, and the north acre was a fumigation study. There was not a single broomrape emergence. We scouted for 12 weeks, several times per week, and none of us could find anything. It's pretty bizarre. We have a lot of questions, which I guess is good as a researcher, but not so good if you're a practitioner looking for solutions. What worked and did not work? I was talking about that late planting date. That was like just the plants looked pretty sick through the whole season. They did fruit and we did get a yield, but that's just, you probably don't want to plant your tomatoes in super late June, followed by a heat wave. The soil had like commercially acceptable tilth, but it wasn't the greatest. There was some weird wetting patterns with the irrigation. We're going to investigate like differences in those alternate timings, because it's been noted by some Chilean researchers that branched and Egyptian broomrape don't have the same phenology and they differ in development timing. So that's kind of what we were trying to get at with those alternate timings. And we'll continue to look into that into future studies. Future research, we're going to do another efficacy study this summer in that same field. We're really going to look at rimsulfuron applied as a chemigation treatment. It's already registered for a foliar application, so that's kind of good luck as far as registration is concerned, but we do need to get data to possibly get an indemnified label or a different application on that label. Planting dates, that was a big hypothesis as to why there was no broomrape. We're going to investigate 3 different planting dates and see how that affects broomrape emergence. I'm currently doing a variety screening for broomrape sensitivity. Again, that was another hypothesis. I'm doing that right now in the contained research facility we have on campus. And I initiated a rotational study in 2021 on campus again to focus on imazamox and see what that does to rotational crops. And hopefully after this field season, we can find some promising treatments and put those into larger scale demonstration studies in 2023. I'd like to acknowledge the Israeli researchers for pioneering this whole thing. And I'd like to thank Gene Miao. He was huge as a collaborator in Mosin Mesgren, Schreiner Brothers Farming for providing the field site and just being great to work with. They did just huge asks and lifts for us. So I can't thank them enough. Ag Seeds Unlimited, Cal Transplanting, and Wilbur Ellis Woodland for material and farming support. And then funding sources for this work included USDA R4 program, the California Tomato Research Institute, and the CDFA Specialty Crop Block Grant Program. And a huge thanks to the Hansen Lab for helping me scout for Broomrape, because it's pretty difficult and we're out there a lot. And with that, I will take questions. And if you'd like to find out some additional stuff on branch Broomrape in CalAg in April, there was an article written by our colleague Wale, and it's a good primer for Broomrape. And again, feel free to reach out to me. We're always looking for collaborators with infested fields. I'm not a regulator. I'm not the Broomrape police, but yeah, I'll end with that. Thank you.
Okay, thank you, Matt. The DPR poll questions are up. I don't know if we need to read them aloud. I don't see any specific questions in the question and answer section. Please post those. I'm sure people, if you have questions, just put them out there. Since we do have a couple of minutes, let's just read those questions for DPR.
Sure. The first question is, Broomrape parasitizes a plant's blank shoots, roots, vegetative growth, or flowers. And the second question is, which herbicide applied during the rotational study injured corn the following season? Imazimox, imazapyr, sulfosulfuron, or rimsulfuron?
Okay, and let's have let people answer. Submit your, remember to press that submit button there. There is one question in the question and answer section. I saw question about when did you were reporting or your protocols were set to have applications at various growth dates, growth degree days. So when did you start the counting those?
We started counting the growing degree days at transplant.
Transplanting, okay, all right. I have a question. I personally, I call this weed a silent killer, because you have no idea you have it in your tomato field before you see it. You see those pretty flowers and then it's absolutely too late to do anything. So what about fumigants? I think you mentioned that you had a study set up last year or this past season, testing different fumigation methods, but that did not really give you any results, right?
Well, yeah, we didn't get Broomrape, so unfortunately we didn't have results. I believe that there was an exemption made for methyl bromide to be used because of its quarantine nature, but that's upwards of $5,000 an acre. So I think, I don't know how many growers are able to do that.
Probably not. What about the products that you can put through the drip, like something like metam, sodium, metam, potassium?
We did include those in that study, and we will be looking into that in the future. We're kind of asking the growers what they think, and that's a very popular idea right now, is K-PAM, metam, all of those things, as well as anhydrous ammonia. Yeah. So we'll be looking at those probably. Yeah.
Okay. So I know we are kind of running into the break time, but I have I'm curious, what is your theory? Why is this old weed showing up again? Is it something that happened with the climate? Is it something that happened with the varieties we are using? Why do we see it again?
I'm not going to be, I can't answer that. I'm sorry. I can tell you, I can tell you the ideas that I hear. Some people say something to do with drip irrigation. Some people say it has something to do with consolidation of the industry. Some people say we're fertilizing differently. I don't know. to be honest. I don't think
Yeah,
no
but
one knows, right?
That's why you are doing your research, right?
Exactly.
Yes.
Yeah.
Exactly.
So I
Okay,
can go over the answers
yeah.
to these questions really quick.
Yes,
Number
please.
one was broomrape parasitizes a plant's roots. It's a root parasite. There are shoot parasites like dodder, but broomrape is a root parasite. And then which herbicide applied during the rotational study injured corn the following season? That was sulfosylfuron. Must have not made that clear. I'm sorry. Sulfosylfuron is outrider. It's a wheat herbicide, and it did injure corn, and that presented as stunting and chlorosis.
Okay, very
Thank
good.
you.
Thank you, Matt. Excellent presentation. And we are ready for our next break, I believe.
Well, hello, everyone, and welcome to the last technical session of the day. My name is Eric Ellison, and I work as a director of agronomy for a controlled release fertilizer manufacturer called Purcell Agritech. I've been on the governing board for CA-ASA for several years, and this is my last year. I will be moderating today's session on biostimulants, and we have an excellent program covering regulatory concerns, plant growth promoting bacteria, and field evaluation of commercially available biostimulants. For speakers, I will inform you when there is about 5 minutes left, so there will be some time for questions at the end. And for attendees, I'll just remind you to select the question and answer tab near the bottom of your screen to submit questions to the speakers. And that question answer tab shows up when you move your mouse. And so you'll be able to see it, otherwise you won't see it. So let's go ahead and get started. Our first speaker of the day is Nick Young. He is Environmental Program Manager with California Department of Food and Agriculture, and he's associated with the Fertilizer Materials Inspection Program. Nick recently finished his term as president of the Association of American Plant Food Control Officials, and in recent years, that organization had formed a biostimulant working group to address regulatory concerns or issues in terminology. And so I thought it would be appropriate for Nick to provide his expertise in this area, given that there may be some changes in guidelines for registering biostimulants. And also there's some recent EPA draft guidance for plant regulator label claims, including plant biostimulants that may be of concern here in California. Anyway, the title of Nick's presentation is Biostimulants from a Regulatory Perspective. And I'll turn it over to him. Thank you, Nick.
Great, thank you, Eric, and good morning, everyone. Let me go ahead and share my screen here, get the presentation up. Okay, thank you very much. So my presentation this morning is a little bit different from what you've been seeing during the conference, and for biostimulants, mine is specifically from a regulatory perspective. So I'm going to provide what you can and can't say at present time, and a lot is changing in this industry. So as Eric had mentioned, I'm with the Fertilizing Materials Inspection Program and also co-chair of the Biostimulant Committee with APFCO. And so before we start talking about biostimulants, I'll briefly go over the Fertilizing Materials Inspection Program and kind of what we do. So we promote safe and effective distribution of fertilizing materials. And that means we perform inspections in the field, we collect samples, perform lab analysis, we investigate complaints, we review efficacy data and labels for registration. And all of that is designed so that growers can feel confident in the quality and quantity of the product, so that you're getting what you pay for as you're growing fruits and vegetables and fiber in California. let's see here. Okay, so we say fertilizing materials because it's not just fertilizer. We regulate a lot of things. So obviously commercial fertilizer for agriculture, but we also regulate organic input materials. So everything that's specific for organic food and crop production. Also what's called agricultural minerals, things like gypsum and lime. Specialty fertilizer, so that would be your home and garden products. Packaged soil amendments. And then lastly, we have a category called auxiliary soil and plant substances, which is kind of a catch-all for everything else. And a lot of the typical biostimulant ingredients would be found within this category. So essentially our program, we regulate everything except for pesticides and for bulk conventional compost, but we have our hand in everything else. So how does that apply to biostimulants? First off, I think there's this helpful slide. It actually looks like it got cut off on the bottom for the credit. I think this originally came from Agrisen via the one of the biostimulant coalitions, but it shows you biostimulants is such a broad category. It in and of itself, it can mean so many things, as you're probably well aware. Some things that can be considered pesticides are in here, as well as things that would fall into our purview as fertilizing materials. And there's very common ones like humic acid, some of the seaweed or kelp extracts, beneficial bacteria and mycorrhizae, and things like soluble silicon and others. So it encompasses a great deal, and why this has been such a challenging category for not just California, but for all states to get on the same page with this. So this may come as a surprise since everyone is talking biostimulants, and it's very, very common on an industry level and on an academic level. But for the most part, in the United States, the states don't recognize the term biostimulants at present time. You cannot have it on any form of fertilizer labeling. And this is with Canada as well. And about a year and a half ago, this applied to all states. But since that time, Vermont and New Hampshire now have laws that they do permit biostimulants on fertilizing materials labeling. And the others don't, but we're working on it because we want standardization and uniformity. So that said, I want to be very clear that we still regulate a lot of the ingredients and compounds that would be considered to be biostimulants. So you saw that slide, all of those things. A lot of those you're able to have on a label, you're able to make claims, and there already exists a path for distribution in California and other states, and there always has been. You just can't refer to them as a biostimulant. And I'll get into a little bit more detail on why that is. So for state regulators, it's critical that the label identify what a specific biostimulant is. And the reason for that is biostimulant doesn't tell you that. And a lot of state regulators view it as a marketing term. So if you were to say, yeah, there's 10 biostimulants in this product, well, what does that really mean? What is it? And to be able to identify ingredients that truly work for a grower versus something that might be snake oil that there's no data behind. So if you have a label, you're promoting humic acid, or I gave some examples here, soluble silicon, Bacillus subtilis, kelp extract, and on, you can still have those on a label and still promote the beneficial effects of them. But you can't say, we have a biostimulant. You have to identify what it is. And if the ingredient is presently recognized by AAFCO, the Association of American Plant Food Control Officials, it's typically permitted on labeling in most states and Canada. And so how does this come into play as far as the biostimulant industry and what their goals are? So for one, they want to be able to use the term biostimulant. If you have these ingredients and some of them really work, and this is now a term that's used regularly, and state regulators know this, throughout agriculture. They want to be able to use it on labels. They want to make these biostimulant claims. They want credibility that there are a lot of these products that work. They want that clear, consistent, predictable process. And very importantly, one label for all states. There's a problem now with a lot of these ingredients that various state laws and regulations across the country that they have to have different labels for different states to abide by some very specific criteria. And I'll also get into that in a little bit as well. And they want to be able to point out dual uses for active ingredients, things that may be pesticidal in nature as well. So they want a path to market. There's a lot of industry involvement about finding the solution, about having uniformity and standardization. And these are just some of the groups that have been heavily involved in this process, from the USDA, EPA, NASDA, FDA, APCO, state of California, and many, many coalitions and trade organizations. So if a biostimulant ingredient isn't already recognized, again, something like, say, humic acid or soluble silicon, it still may be possible to have within a product label to distribute in California and other states, but either that ingredient needs to be formally recognized and defined within APCO's official publication, or efficacy data is required. So if it's not defined in there, that may be fine if there's robust data that our scientists can evaluate to say, yes, this does look like this particular ingredient or compound works, and it can be used throughout California and it's safe. So even if it's new, there are always paths towards distribution. So if an ingredient is recognized, if it's a nutrient, it'll be in a guaranteed analysis on the label. So that label is guaranteeing a minimum amount of that nutrient. So a grower knows what to expect, and their advisors will be able to know how much their crop is getting of a particular nutrient. Or if it's non-nutritive and something along the lines of a biostimulant, it would appear in California, what's called a non-plant food ingredient statement. Other states, it may be a little different. Also give examples of that too. So we're right on the precipice of a lot of movement, I think, with biostimulant uniformity and some decisions finally moving forward. So in less than 2 weeks, the Biostimulant Committee meets in Mobile, Alabama. And this is at the Winter Annual Conference of APCO. And there's a lot on the table. There's been a lot of working groups focused on very specific things that hopefully, optimistically, will be moving forward. One of which is a formal definition of what a biostimulant is. Another is uniform labeling, something that works in all 50 states and is simple and easy to understand. Efficacy data guidelines. So this is standardization towards, if you provide efficacy in California, will that same efficacy be reviewed and approved in Florida and evaluated the same way? That right now is easier said than done. So some states have requirements that the studies have to be performed at universities within their state. It makes it very problematic for particularly smaller companies to be able to do that throughout the United States. So we want to eliminate that barrier, have just general guidelines that all states will accept, as long as it's similar conditions and soil types, that we can have firms submit to recognize some of these new novel ingredients. We're also going to be talking about a model bill, which is a comprehensive thing to present to state legislatures in order to create uniform laws across the United States. And then lastly, looking at laboratory methods. And there's many that are already established. So we already have lab methods for, say, humic acid and soluble silicon. But if you have novel ingredient, say it's a botanical extract, there may not be a known or recognized method for that yet. And you have to be able to quantify it. We have to be able to prove to a grower what you're saying is in a product is actually there in the quantities that you're stating that it is there. So all of these things are going to be discussed. We've had working groups working on these, as I said, the last 6 months. I think that there will be a lot of movement on the first three. And I'm going to provide some updates on where we're going with the definition and the uniform labeling right now, because it will affect growers in California and what you guys will be seeing in the future for these products. So there's a lot of iterations in the past of what a plant biostimulant is. The EU has their own definition. There's definition proposed in the 2018 Farm Bill. Multiple definitions in the 2019 report to the President and Congress, and ISO is looking at definitions. The EPA does not intend to develop a separate definition from USDA. So the one that's gaining, I think, the most traction and uniformity right now is in that USDA report to the President and Congress. There is what they refer to as alternate definition number 2. And this one is the one that APCO is currently looking at to potentially have standardized throughout the US. And it is a plant biostimulant, is a substance, microorganism, or mixtures thereof that, when applied to seeds, plants, the rhizosphere, soil, or other growth media, act to support a plant's natural nutrition processes independently of the biostimulant's nutrient content. The plant biostimulant thereby improves nutrient availability, uptake, or use efficiency, tolerance to abiotic stress, and consequent growth, development, quality, or yield. So this is what will be discussed within the next couple weeks at APCO. I don't know if this is going to go through as is. I know from state regulators, one of the concerns is this, the last sentence is very definitive, that it thereby improves these things. I think from what I hear, they want it softened a bit to be may improve, because other factors factor in here. So for instance, soil pH may matter on effectiveness, or moisture, other content, quality of soil. So to say that it will definitively improve isn't necessarily always the case when you look at data. So this is close, and this is essentially what we're hoping will pass, but we will know where it's going within a couple weeks here. So now I'm going to go to labeling and kind of show you the mess that everything is currently in and how we want to simplify things moving forward. So this is the California example. Again, we don't recognize biostimulant, that term, but the ingredients we allow on labels. And if this had fertilizer in it, and this example would, it would have the fertilizing ingredients under a guaranteed analysis. However, underneath that, you would have this statement that says also contains non-plant food ingredients and the percentage of these biostimulant type ingredients. I'm not a big fan of the terminology non-plant food ingredient because it's not the most elegant term, but California was actually one of the first to, or maybe the first, excuse me, to recognize these, and it originated back in August of 1991. So this is how labels have been in California for 30 years now. But other states developed their own subcategories, and this is why the importance of a 50-state label makes a big difference. There's confusion with the industry and growers with all these varying label headings. So you have ours, but you also have beneficial substances in some states, soil amending ingredients, plant amendment ingredients, active and inert, and total other ingredients. And so to be compliant across all these states, you have to have a lot of these headers for the same thing, the same message, and it makes it very, very confusing. So here's an example of that. This is bureaucracy at its finest. So this would be a present day, quote, universal label for a fertilizer product that also has these biostimulant ingredients in it. It's what is this? What is this saying? It's very confusing to a grower, very confusing to industry. But this is what's needed if you want to be across every state, and it's frankly a little ridiculous. So at AFCO, we're striving to deconstruct this, go back and just keep it simple. So the proposal that will be coming forth in 2 weeks is the next slide, which is a lot more elegant. It is simply that also contains beneficial substances, will most likely be the term, and then you have the ingredients after that. Now, the change from non-plant food ingredient to beneficial substances, we had a working group of about 50 people comprised of regulators, fertilizer industry, university researchers, and on, scientists who work in labs. And I was hopeful that there would be a firm consensus of one over another. We had a poll. I went to check the poll, and it ended up completely 50-50 down the road, down the line between beneficial substances and non-plant food ingredients. So after a lot of negotiation and further discussion, we got some consensus. It appears to be, and it will be beneficial substances. The reason why this is not biostimulants is because there are ingredients that fall into this category that aren't actually biostimulants. So things that would be maybe polymers, some wetting agent products that wouldn't fit the typical definition of a biostimulant. So beneficial substances is where these would go. And as long as we have a definition approved for biostimulants, you'd be able to make biostimulant claims within the text of a label. So I'm going to touch on lastly a few slides on the pesticide angle. Now, I don't deal with pesticides, but when you're dealing with this biostimulant issue, there's a complete crossover. So products like these with biostimulants, they can require dual registration as both a fertilizing material and a pesticide. And these can be based on the claims on a label or the composition. So ingredients like some microbes and seaweed extract may possess properties that may enhance nutrients and soils, but they also act as a pesticide or a growth regulator. And so just because a product is registered with us or with DPR as a pesticide, it doesn't preclude additional registration. A little over a year ago, the EPA released an updated draft of their guidance for plant regulator products and claims, including plant biostimulants. There's a link to it here, so when this gets posted later, you guys can go to it if you're not familiar with it. But what the EPA is saying is that the claims are a key consideration to determining if a biostimulant is a pesticide or not. And it's claims-based, but not claims-only based. Product composition is another consideration. Products that are not considered pesticides are plant nutrients and trace elements, plant inoculants, soil amendments, and vitamin hormone products. And in that document, they have a bunch of tables. The tables of 1A through C are examples of these categories. Table 2 is some generic non-pesticidal claims. And then here are some examples of plant regulator pesticidal claims. So if you're stating with a biostimulant that it accelerates or retards the rate of plant growth, maturation, or otherwise alters behavior of plants, it would be considered a regulator claim that would be pesticidal claim. And all of those are within table 3. So here's an example of these. And this is where we kind of get into some crossover now that is maybe problematic. It enhances or promotes, stimulates fruit growth and development, or plant growth and development. Some of these are very similar to what you commonly see with biostimulant claims. Just to provide a comparison, and I'm not going to read this whole thing, but this is our definition in law with the Department of Food and Agriculture for this auxiliary soil and plant substance category. As you'll notice, it's very similar to some of those prohibited or pesticidal claims that would be accelerating growth and things of that nature. So I highlighted some that it's intended to improve product quality or other desirable characteristics of plants. If you go back to the definition, the pending definition, the plant biostimulant thereby improves development quality and quality. So there's some crossover there. EPA provided specific examples of biostimulants that may be problematic for them, and these are going to be big challenges. Corn gluten meal, which is not permitted. Humic acid. So with the EPA, the foliar use would not be permitted. It would be considered to be pesticidal as a growth regulator. That's an issue. Obviously, in the Central Valley, we have a lot of that type application. And then seaweed extract, depending on the claims, and if they're making claims related to naturally occurring hormones that would be in seaweed extract, it may cross over into that territory as a pesticide. So
we have about 4 minutes, Nick.
Okay, great. Yeah, I'm just finishing up now, and we'll go to some Q&A here. So they had stated that regulatory approaches for products that have multiple plant regulator and non-plant regulator modes of action, if it can be demonstrated that a particular product has the activity claims on the product and does not make any plant regulator claim on a label, it may be excluded from FIFRA regulation. So for these seaweed extract products in particular, it's rather delicate dance of what claims can be made on a particular product. And that's it. So I think I got enough for 3 minutes or 3 or 4 minutes for Q&A. So I'm ready for questions.
Very good. Thank you. So the first question we have is, how will CDFA define when an ingredient is beneficial? How does this tie into efficacy? I know you didn't really get into any sort of efficacy discussion, and I think that is another part of the working group and what they're trying to define, but you can go ahead and try to answer that live.
Sure, absolutely. So if it's an ingredient that is already known to have beneficial use for plants, so say for example for humic acid, there would have to be no efficacy submitted. They would just be submitting their labels and accompanying documentation for our review, and we would accept claims for that particular ingredient that have been accepted. So in the case of humic acid, it may increase nutrient uptake, so we allow that claim on labels. However, if it's novel, if it's something that's new, say a botanical extract that we've never heard of, then they would submit the data to our scientists for review to whatever their intended effect is. They think it does XYZ, they're going to submit us the data for review. And if the science is sound, we'll approve it and we will allow it on labels. If not, we'll tell them what we view the deficiencies are in that data and request that they go back and look at the missing parts.
There was a second question. How do you deal with verbal, but not on the label pesticidal plant regulator claims? So I guess this is maybe the wink, wink, nod, nod part of biostimulants.
Yeah. It's that one is the biggest challenge, obviously, verbal claims, because they're not allowed to make those, but salespeople do that all the time, right? So in our law, any claim you make, and this would include verbal, constitutes labeling. So they aren't allowed to say the things that aren't in our laws and regulations. So what we commonly deal with is websites. So on the label, they'll have a very clean label that doesn't say prohibited things or make over promote their products. But on the website, then they will say a lot of those things. And we will issue, if we're aware of it and know about it, we will either issue them warnings or violations that they have to change that. Now, verbal, we very rarely come across that. unless a grower or some other entity files a complaint with us that says, I have this email that said that this product would do this, this, or that. And then we can follow up with it and do an investigation.
Well, I guess just finally, very quickly, there was one comment in the chat that stated, obviously, biostimulants will skirt the EPA requirement for efficacy data. So I guess maybe the question is, Nick, how will any of these efficacy guidelines change going forward very quickly? If you could just give us a quick synopsis of maybe what the working group is migrating towards on that.
Sure. So I think on an individual ingredient level, it's a very robust system, both through California and through APFCO, in order to get this a new ingredient as a biostimulant recognized, right? So all of that efficacy is evaluated, and it won't be approved if it's something that is very conditional, like only works on one particular soil type or lab type conditions, but not in the field. So if it makes it into being defined, it means it has gone through a rigorous process. But there is also a lot of snake oil out there. And that's why it keeps getting kicked back. For all this data that's submitted, there's a lot that's problematic that's kicked back. And in effect, they're not allowed to make claims of certain ingredients because there's no proof that certain things work. And so that's what it is moving forward. It's just continuing to have this robust system in place. And the part two of that is having it be standardized for all states so that they all have a baseline of what they're supposed to do when they're looking at this efficacy data and how the trials are supposed to be run and how the data is supposed to be interpreted. And that's currently being evaluated.
Very good. Well, thank you, Nick, again for your wonderful presentation. And we look forward to seeing what comes out of the working group, what decisions are made at the next AFCO meeting.
Yeah, thank you, Eric. And yeah, there'll be a lot of new news, a lot of things moving forward in the next couple of weeks.
Great. We'll look for that. Thank you.
You're welcome.
Our next speaker is Dr. Ann Hirsch. She's a distinguished professor of molecular cell and developmental biology at UCLA. And today, Ann will be describing some very interesting research on promising beneficial soil microbes and the scientific rigor, I think, involved in the discovery process. And I think we're going to get a big dose of hard science here, and that's good. We can all use that in the biostimulant world. And I'm delighted that Ann agreed to speak at our conference. The title of her presentation is Soil and Plant Microbiomes, Complex Ecosystems that Harbor Novel Plant Growth Promoting Bacteria. Take it away, Ann. Thank you.
Well, number one, that was the old title. I have a new title now. And partly because we've added another parameter. But let me just go ahead and begin to talk about what we do. And first of all, I want to thank you all for inviting me. This is a great pleasure for me to talk to you. And what happened is we had some really great talks yesterday about water and about how California is drying up and what the different procedures and ways we're thinking of sort of trying to improve it. And I think this issue in science in 2020 really sort of brought it into focus in California, looking at essentially NASA's GRACE satellites and looking at the water masses and how they've changed over the years has brought this up in very strong focus that we are in trouble in terms of the amount of water that's left in the state of California. So our goal has been to find microbes with plant growth promoting properties, which is not the same thing as biostimulants, I understand from the first talk, and I appreciate that. But what they're looking for things that can help plants grow in a hotter, drier California. And generally this will include nitrogen fixers, and like rhizobium and azospirillum, phosphate solubilizing bacteria like bacillus and various actinomycetes, and many, many others. And so what we decided to do is look at different sites in California and some not in California to find whether or not we could find bacteria that could sort of fill these new roles. And so we started actually about 10 years ago now, almost 10 years ago, in two places, in Israel, in the Negev Desert, in a collaboration with Drora Kaplan, and in Botswana, in a collaboration with Dr. Pauline Muhlenberg. And these two areas have very, very extreme periods of dryness. But yet, when we look in the springtime, we can see that there's robust plant growth. Many of these plants are legumes, but specifically in Israel, there are all kinds of legumes in here because I still have maintained a very strong interest in nitrogen-fixing plants. But the dominant plant I'm going to be talking about here is Zygophyllum, which remains green in the incredible different circumstances. But just to give you an idea, one of the things we were concerned about, especially as we have a lot of undergraduates in the lab, these bacteria can be anything. And so what we try to do is follow a procedure for biosafety. And what we start out doing is usually collecting the soil, or in some cases later on, we're going to be isolating the bacteria from plant tissues as sort of a winnowing effect. But what we do is we mix up the soil and we do metagenomic analyses. And we heard about metagenomes yesterday. But the idea here is to get what kind of bacteria are present in this soil. And what we then try to do is we try to culture these bacteria. And as you know, probably about 3% or 4% of bacteria are culturable. So we get a very small number of bacteria that could be used as inoculants. We identify them using 16S RNA. And then what we also start doing is start asking the questions, well, what do they do? Sometimes we can tell by the name of the bacterium, what it could be, like if it's a rhizobium, that it's a nitrogen fixer, but not always. And so what we want to do is learn what they are, find out what chemicals they're going to produce, so we can get some idea of how the bacterium actually affects the plant, such as like phosphate solubilization or siderophore production or other types of production of different kinds of lipopeptides, which can kill fungi. And so we test a lot of these always on plants. And to be sure that it's safe, what we also do is we test things on plants to see they don't kill the plants. We test them on nematodes, and we use Caenorhabditis, C. elegans as the nematode of choice, because that has been used classically to show pathogenicity from bacteria or not. Pseudomonas aeruginosa is the bacteria that kills, and what we look for are things that don't kill. We haven't done mice because mice are expensive. But then we go on and we do DNA fingerprinting so we can learn other things. We look for whether or not the bacteria have the type 3 or the type 4 secretion system. So we use the genes often to tell us also what the bacteria is, but what is it safe. So here's an example of the kind of assays we do. This is the worm assay. So these nematode worms, if we look at comparing Pseudomonas aeruginosa, you'll see under the different time periods, all the worms are dead. But if we use its own food, which is E. coli OP50, by 120 hours, everybody's living, everybody's very happy. Now, the bacteria I'm going to talk about first is something that we isolated from Israel, and this is Deutzia cinnamomea, and we call this strain 55. And what you can see, it's not exactly as, at the same level, because Deutzia is probably not as tasty to the nematodes as E. coli. The E. coli has been its food for as long as we know. So what we do next is we then do the plant studies. And what we can also see, this is the responses. In this case, the pathogen we're using is Burkholderia cepacia, which is also not only kills plants, but is very not very good for people. And you can see there's no effect of the Deutzia cinnamomea on the different plants. And we've done these in other plants besides alfalfa. But the assay that we just introduced recently was using waxworms. And what you can see in the case of the control, the worms are moving around, they're reproducing. The same is true for Deutzia, but with Pseudomonas aeruginosa, we see all these dead melanized waxworms. So we then take a look at the genomics. And so we did this in collaboration with the Joint Genome Institute as a part of the community sequencing project. we found no obvious virulence genes. And as I mentioned, the type 3 secretion system is one of the worst because it introduces something into the cell. A type 4 secretion system is like in Bordetella, whooping cough, but we didn't see any of these. But since this is a gram-positive bacterium, it's an actinomycete, it does have a type 7b secretion system, but this is very, very different from the sequencing of the pathogen of the secretion system of the pathogen strain. So how does the stress tolerance occur? We think this is one of the mechanisms that I'll show you the data in a minute, how plants will survive when the seeds are coated with Deoxycinnamia. And trehalose production is one of them. There's all kinds of different transport systems. We have all kinds of proteins that are expressed, and of course siderophores as well. So we mine the genome to find as many strategies as possible. And this work is published, and so you can see this, the references below. So the most exciting thing was we started to grow corn, and because we wanted to test again on a plant. That's part of our strategy. And for some reason, we chose corn. I don't remember why. But we did these experiments both inside the greenhouse and outside. And what we saw was a surprising increase in corn production and compared to the controls. And this is especially obvious if we look at the cobs. And we have all the, the data on this. This has been published, so you can see it there. What we did next was collaborate with Heinz Hoben at a small company, inoculant company called BioNext. And what they showed using these corn trials was that there was an increase, a significant increase in both 2019 and 2020 in the growth of the corn. And so this was very exciting to us, and this started us thinking more and more about looking at plant growth promoting bacteria and their possibility as replacement for many of the chemicals, synthetic chemicals that we're putting onto our plants. Now, in this 2021, it was a lot of rain, and so the cornfields didn't do well, but a preliminary soybean test showed that seems to be an effect as well in the production of this is the number of nodes that are on the stem in the soybean plant, and this is the amount of yield. So there is the increase that we saw also with the corn plants. So more recently, we've started to look at wheat. Now, wheat is, again, a very important grain in the United States, but it's also an extremely important grain in many other parts of the world. And here again, we see an incredible increase of the wheat kernels relative to Deoxycinnamia. And this is a wheat variety. We had a project working with people in Egypt, and so this is an Egyptian variety. And so in collaboration with David Still of Polypomona, we're going to start looking at some American wheat varieties. So these are more or less in progress. These are recently, we had the wheat grown outside during the winter in California, which is a little bit warmer. And you can see again, there is not as good as the first time, but we also had a lot of predation from critters. Interestingly, not so much in the Deetsy as was in the control. And so we'll be repeating that experiment. So in Botswana, what we did here is we took a different strategy because we wanted to sort of see what does the plant select? What does a plant take along? As you know, in the human microbiome, you get your microbiome from your mother in a vaginal birth. And so the human will select certain bacteria that will establish that microbiome at an extremely early age. So we're curious, could we establish a microbiome in a nodule? Because this is a very discrete bunch of cells and they're fixing nitrogen. Could we actually see what this was? And so we collected the trap plant. These were trap plants. This is the normal plant. This is Tephrosia purpurea, which is grown in a farmer's field in Botswana at the Botswana University of Agricultural and Natural Resources. The soil was sent to us with permits, and then we use that to inoculate cowpea plants. And then we would isolate them after surface sterilization. And this is what we now are looking at. We're looking at culture independent analysis. We're looking at the organisms that are present in the microbiome. But not all of them are really there in very large quantities. You can see this is sort of the list that shows you who's there. But the big players are the prokaryotes, the firmicutes, and the actinomycetes. By the way, Decia is an actinobacterium. So many of these are already known plant growth promoting bacteria, but we reasoned that these are the ones that are most likely to be inoculants if you were going to isolate them either from soil or from nodules. So now what we're doing here is we're taking a look just at the rhizosphere of the Tephrosia. And this is, again, the soil eDNA data. And the proteobacteria, the firmicutes and the actinomycetes and this fourth group, they're there, but they're in different ratios. So we think of this as sort of like, going when you have a huge highway and you have to get off and you come down to a single dirt road. This is really winnowing the number of DNA sequences that are available. And this now shows us the conundrum. If you isolate the bacteria from the nodule or anywhere, what you'll see are just a few bacteria because of this essentially block in the number of bacteria that can be cultured under artificial conditions. So this shows you the cowpea nodule microbiome. And in this case, the vast majority of the bacteria are rhizobium. And this makes sense because that's what his job is to fix atmospheric nitrogen into ammonia. And so it's 95% rhizobium and bradyrhizobium. And then you have this little group here. And this is where we began to focus our interest. So I list here of the cultural bacteria shown. And one of the surprising things, besides all the great diversity of different types of Rhizobiaceae, were in the number of Methylobacterium. Now, methanotrophs are very important, and they're turning out to be, especially the pink pigmented methanogens are turning out to be also plant growth promoting bacteria. But there's a diversity of other things, and many of these are even human pathogens. And so this is one of the reasons we really do all this biosafety work, to be absolutely sure that what we're working with is safe. Now I'm going to switch to talk about what we did at Cal Poly Pomona. And this was because they had, at that time, large numbers of alfalfa plants growing there. And so with my postdoc from Spain, we decided to collect the nodules of alfalfa from this big field. This is Ethan Hum, who's the second author, and as I mentioned, this is in David Still's group, garden, as it were. And again, what we see is this difference. And what we also did, which was a little bit different, was not only look at alfalfa, but we went to the Mildred Mathias Botanical Garden to look for Medicago polymorpha. Medicago polymorpha is actually a weed in California. And so we looked at both genomes, and I'll just show you the difference between the nodules here. This is, first of all, this is the sativa, Medicago sativa rhizosphere soil, and actinobacteria, proteobacteria, firmicutes, but in this case, acidobacteria. So we've got a slight variation compared to the cowpea. And if we look at the nodules, and we see the vast majority of them are rhizobium, and there are some very few other ones that we are not studying as much at this time, but we will take a look to get an idea of what else is in the nodule. So what we did find is a few of them. And these are now alfalfa plants that have been co-inoculated with the symbiont and with certain strains that are the nodule inhabitants. Bacillus, of course, and what we are doing is a double inoculation with the idea of potentially developing a consortia whereby one could, like almost having like a fecal transplant for plants, but using bacteria that are from the soil. And what we can see is incredible changes in all these things that are darkened like this, are bold-faced, are showing you the increases in growth. This is another actinomycete. This is Micromenospora. And routinely, we have found that this is an excellent plant growth promoting bacterium. It's a filamentous actinomycete, making it more difficult to work with, unlike Decia, which is not. And then we see this bacterium. This is why I changed my title, because of this bacterium here, Variovorax. Variovorax doesn't do anything for plant growth, but it does something that may be more important in our future times. And that is, it seems to be very good for working on bioremediation. Variovorax, or various feeders, is the name, is what it means, is essentially able to degrade all sorts of things. And this bacterium was studied by Jeff Dangel's group, and there's a paper in Nature about them by first author is Finkel, Finkel, and what he stated, and what they state in this paper, is that the Variovorax is sort of fundamental to root development, and they have a lot of beautiful work that shows this. So the three that we isolated from alfalfa nodules are Variovorax paradoxus, two different species, and Variovorax baroncumulans. And what we did is we started growing them on heavy metals. And we did a number, but in this case, we've only have just showing you the copper and nickel. And what we see here on alfalfa, and you can just barely probably see the asterisks here that point out the statistical differences, but we can see that there is a statistical difference, and such that the Variovorax paradoxus in this strain and the Baroncumulus can actually considerably help plant growth over the uninoculated control. Similarly in wheat, and in this case we took some other isolates. These are the Variovoraxes here, and these are some other isolates that we've looked at, and here we don't see much effect. But the Variovorax, again, help with not only promoting growth with just in the control, but they are also effective in keeping the plant from succumbing to copper toxicity. So these are pretty preliminary data, and I'm going to just let it stop there, because one of the things we know in LA is that there's a lot of contaminated soil. There's a lot of brownfields, for example. And so what we really were thinking about, could we use something like Variovorax or all these different bacteria that plants have taken up into them, into their tissues, in order to make more food for people to grow with less damage to the soil? Because one of the important things that we keep forgetting about is soil, and how crucial it is for us to produce the food that we have. And I like this quote. It says, "If seeds waited for the perfect conditions to grow, there would be no plants in the desert." And that is true. Plants will grow in all sorts of conditions. But I think also we have to produce plants growing in agricultural situations and wherever else, so we don't damage the soil, because this is a precious resource that we cannot afford to lose. So thank you very much for your attention. I thank all the students who worked on this project, and the DOE, and we've had funding from many agencies, and the two that were especially helping in desert studies are listed here. And I'm happy to answer any questions.
Thank you, Anne. Very exciting work, I must say. Those are some interesting results from soil inoculants. So I guess there are no questions yet, but I have a question for you, and that is in regards to, I think you mentioned it in your talk, and that was when you choose a particular strain, are there any criteria that you know of that would make that strain better able to compete in the soil or as a seed treatment? Or is there anything like that you're studying that would help to say, yeah, we could use this particular strain at the field level or in a soil, and it would be able to compete with the native bacteria? Or maybe it doesn't need to compete. Maybe it's just associated with the plant.
I think in a way, if you're going to use an inoculum, especially on a seed inoculum or something like that, it will outcompete what's out there. But the question is, we really don't know at this point what traits are important for survival in the soil. I mean, when we looked at things like the desiccation resistance, that was an attempt to start going in that direction. But so many things are really, there's also this sort of almost co-evolution between certain plants and certain bacteria. So this is a very good question, but it's a very difficult one for me to answer because I think it is really complicated. And
Well,
frankly, there's not funding for it, to actually even do this kind of work, because you'd have to really start getting the genomes and doing a lot of experiments and trying to get really, it would be a big data push.
Well, you did show some positive results or some benefits from inoculation. So I think, that's a step ahead of me and some of my experiences with some of these products in trying to inoculate. But, that to me, I was very surprised that you were able to see something in the field like that. That's a great result.
Yeah, well, this bacterium has actually never really been looked at all. And that's why we did so much. There are some instances where, every bacterium has a dark side. And there are some DCs which have been listed as sort of pathogenic, but as mild ones, usually in immunocompromised people. And this one is not, because we did not test, when we did it, we don't test people, that's not good. But we do test the tests we have. And by looking at the genomes, we could also see no evidence for it. So it's a long, laborious process. And this has not been pushed, I think, as much because we had other ways of doing things in the field before. But now that biostimulants are becoming more, of interest, and as they should, because the soil needs the microbes need the soil, and the plants are. It's essentially a triangle connection.
I guess we have time for one last question, and the last one is: what do you mean for the microbiome? That is, for the proteobacteria. Actinobacterial quantification, were they isolated from the rhizosphere soil or from the plant tissue? Are they present on the surface of the roots or within the plant tissue?
Okay, that is actually a very good question. When we isolate soil, that's different. So when we get all these different bands and you see all these different colors, that's from soil, and you just are taking the soil water, and then you isolate the DNA from that material. So that's going to give you a really large quantity of DNA sequences that have to be analyzed. When we start looking at the nodule, and I said it was a winnowing thing, a lot of it also depends, on timing of the plant. So what we're doing generally is doing an averaging, because we're collecting nodules of different ages and trying to get as many as we can. And but overall, the narrower you make the hole, the fewer the bacteria that are come through it, okay? So this technology is going to need more work. And I thought Peggy Lameau's talk yesterday about doing the transcriptomics and thing was another way to think about it. But at this point, I think just the fact that we don't even really recognize how many bacteria that are out there, and the fact that they can't be cultured is another problem. Remember, we're only culturing about 3%. And so anything that's used as a biostimulant that's microbial, it's one of the rare ones.
Well, I guess in the interest of time, we'll have to move on. And I would ask Anne that there maybe are one or two questions in the Q&A that you can answer offline, and that would help get those answered. So thank you. Our next speaker is Michael Rethwisch. He is a farm advisor with the University of California Cooperative Extension in Riverside County. I believe Michael works out of the Blythe office. And I would just mention that once I drove through Blythe in July and it was 118 degrees Fahrenheit. And if there was ever a place to apply a biostimulant for managing heat stress, I think that would be it. That would be where you want to do it. That would be where you would want to run an experiment where you would deliberately impose heat stress. Blythe would be where to do it. So in Nick's earlier talk, he mentioned that the intent in using biostimulants is primarily to enhance nutrient uptake and manage abiotic stress like heat, drought, salinity, and cold. Proving that happens at the field level is very challenging. I think Michael has taken on this challenge in a methodical way and has arrived at some very interesting results, which I believe is what he will talk about today and why I asked him to speak. So the title of Michael's talk, I think, is Biostimulants and Factors Affecting Crop Responses. So Michael, thank you.
Hopefully you can see what I've got on the screen here where it says this one grower said in an advertisement, he didn't do anything different to my grapes than I have done in the past few years, except for applying product X. He doesn't know what it does, but it saved his grape crop, and he's a believer. He'll definitely use it again. Now, as a scientist, this kind of is kind of disturbing in a way, because number one, he doesn't know what it does, and I'm a little questioning if it saved his grape crop, what did it save it from? Good question. And did he have a control to look at it, compare? So we're going to look at some scientific things here, and as I think we know on some things here, there's not everyone agrees on what biostimulants and growth enhancement products do. Some people, as Nick said earlier, some people call them kind of a snake oil. Now, if you've looked at snake oils, they're kind of expensive things because getting oils from snakes is not a good thing to do sometimes. It's kind of tough, and it used to be pretty expensive. But if we look at where we are, look at my comments I've heard from other people, these products don't result in statistical increases in yield. Therefore, it is statistically the same as doing nothing. But if we look at it, some of the growers will say, well, it gives them the consistent economic return, and that's the statistic they like. So the question is, do these things work? Do they not work? Or do they just work some of the times in some of the places? And that's kind of where we are on some things here. We see that we're kind of a rock in a hard place on some things. Some people say they're the greatest things ever. Other people are going to say, no, we're just going to wait till things die and we'll come up and pick up the pieces. And we're going to look at different things. So the question really is, as we look at biostimulants here, is, let's go and get rid of some of the mystery of some of these things and look at the experiences from the field experiments and really kind of determine what the real facts are here when it comes to biological plant growth regulators and the factors that affect those crop responses. Now, my definition of a biostimulant is a little bit different than some of the others. I tend to be more of a purist. I stay away from the inoculant type products because there's who knows what's happening there. So I tend to use a more purist definition, which is these are molecules or substances derived from a biological system or source that elicit a positive growth response in a different organism. It could be a hormone, it could be an extract, it could be a molecule. They could obviously come from bacteria and things like that, but I'm looking for the actual extract versus the bacteria itself for my definition. And Nick's talk this morning talked about some of those things as well. So if we look at biostimulants, there's really three main areas that affect plants. Stress mitigation has been talked about. Some of them we want to see initiate plant defenses, an SAR type response. And some of them we want to see increased plant yields and flowering and things like that. Bottom line, the growers want to see things pay. So if we look at this, if we look at where we are in the United States, probably at 97% of our crops in the United States are agronomic. About 3% are fruits and vegetables. So there's lots and lots of opportunities out there for these types of products. But the question is, and I think Nick has referenced this a little bit, is with so many crop acres, why aren't biostimulants widely used? Well, part of it is, do they work? Do they not work? And I think to understand that, we need to know which is faster and easier to do? I'm going to give you two scenarios to look at here. So is it easier and faster to build something? Or is it faster to destroy something? And if you think about it, I come from a pest management background, and I will definitely say it's, for those of you in construction, it's a lot easier and faster to destroy something than to build something. And from a pesticide aspect, if we think about pesticides versus biostimulants, they're really on two opposite ends of the spectrum here. With pesticides, we're trying to kill something. With biostimulants, we're trying to make something grow. And I can tell you from my work in pesticides, it's much easier to kill or prevent or reduce growth with another aspect with the pesticide, probably 10 to 20 times easier than it is to increase the growth and yields with biostimulants, just because of the multiplicity of systems and steps involved in plant growth. Everything has to work together to see a yield at the end of the year. So if we think about where the EPA is with some of these things, and we've got the 5 very powerful classes of hormones. We've got the cytokinins, the abscisic acids, the gibberellic acids, ethylene, and the auxins. Those have been known for a long time. They're very, very powerful. And most of the products we have out there in agriculture these days are registered, and because these things can have very powerful effects. And they have lots of different aspects on things on the plants that cause them to grow. We know gibberellic acid obviously can cause sizing of grapes. It's used widely in California. It's probably the most widely used what I call plant growth enhancement product, but it's a very, very powerful aspect. But if we look at what is coming out there in this day and age for biostimulants, they're usually what I would call the minor classes of the hormones, the brassinosteroids, the oligosaccharides, the polyamines, the CPPAs, and some of the other things that are out there as well. And these tend to be very much result in very weak plant responses to these chemistries in these what I would call the minor leagues of the plant hormones. So we've done lots of trials over the last 20 years, and the picture we have right here is what we see. This happens to be a smooth brome. It's a forage grass. We've got about 10 different biostimulants out there, and you can see hopefully on your screens that some plots are doing very, very well and had a very rapid growth. And with a lot of products, we saw nothing at all. And I think our take-home point we have from this experiment is that just because you have a biostimulant out there, not every product is going to work or elicit a plant growth response in every single crop. They do have different aspects and different things. So let's take a look at what we have out there. So let's look at the experiences and the facts, and let's just look at something like gibberellic acid, okay? Right now, well, maybe beyond this right now, but there used to be 126 different forms of gibberellic acid that were known. Some of these were strictly in the labs, but if we look what was commercially available, there was GA1, GA3, GA4, GA7, GA10, and some combinations of those things. And GA3 works differently than GA4, and you see different responses with that. So even among the classes, you have different molecules that do different things. So if we look at what we have here, just because you have a product that works, there are a lot of factors that get in the way of making it effective. In this particular picture, this is a gibberellic acid GA3 product applied to smooth brome, and all these different plots that show differences in growth, these are all the different surfactants that were used with it. And so the surfactant in this case makes a big difference into how much gets into the plant to see if there's a plant growth. So if we can't get the product into the plant, we're not going to have so much growth. So just the surfactant can be a problem. we're also going to see sometimes there's lack of nutrients that can cause these things to happen. So if we look at some of our nutrients here, we have 3 different things. We have a low yield, a high yield, and a very high yielding situation here. And as you go from left to right on each of those 3 sets and different products and combinations, we don't see much growth at all or much response in the low fertility area. But if we get the medium fertility and the high fertility, we can see these situations happen. So even if we can get these things into the plants, we will see that if the nutrition is not there, the plant does not respond. So now we've got multiple factors involved at the same time. And here's another graph of that. It kind of shows what we see, where we have, as that fertility changes, the crop response changes as well, just between the treated and the gibberellic acid treated smooth brome here. And you can see we had some sizable differences within 4 weeks. But let's talk a little bit more about California. In California, we've been looking at different products in different combinations. We've been looking at onions. We've been looking. Some people have been looking at some of the peas. And the black-eyed peas, this is with the ProGib 40. Rachel Wong did these up at Yolo County. And we can see that if we look at our yield, we got a 13% yield increase, but in this case, it was not statistically significant. Even though we had a nice trend for increased yield, it did not necessarily come into a difference as far as statistics go. With seed weights, we're about the same. And if we look at some of these other things, we see similar products as well. There are some certain crops respond very well. Again, the lima bean, again, about a 17% yield increase. The bean size, the seed size, about the same. So it's telling us it's not necessarily from a bigger seed, it's more seeds being set, which is what we want to see in some things. But let's look at some of these other biostimulants, get away from things that we know are powerful, like the brassinoster, away from the gibberellic acids, and look at some of these other weaker ones. And we can see that some of these things can trigger growth of soybean roots. We can see some nice early growth, which should help, we would think. And we can see, as we get in mid-season, we can see lots and lots of different pod numbers out there. But by the time we got to the end of the season, we basically saw no yield differences whatsoever. Again, this was a factor where we set the plant up for success, but did not have the yield components as far as fertilizer to bring them home. In this particular picture, hopefully you can see, we have soybeans, and you see some yellow streaks across it in just a certain part of the field. And these yellow streaks are actually where the biostimulant treated soybeans were. And again, they're yellow, not because they had better growth, but again, they turned yellow sooner because they did not have the nutrients in this particular part of the field to express an yield increase. So we have those kind of factors involved as well. One of the things we found very interesting, and we looked at different situations as to why are we seeing differences, is what's happening with the environment. When we did a number of trials, and it took us about 7 field sites over about a 3-year period to get to this point, when we looked at rain-fed fields, we got a nice statistical increase, a statistical value there for rain-fed situations. But when we used biostimulants in irrigated situations, it was not statistically solid and valid. The reason being is that, as far as why, well, a larger root system earlier in the season is going to help with a rain-fed condition because there's more stress. In irrigated, that stress is eliminated. Yield-wise, there's always a slight increase, and it may have paid in these situations, but there's more things out there. And I know we're running kind of short on time, but we'll try to get through this quickly. We looked at products like a twerk, which has a signaling molecule. We looked at it across multiple aspects. In corn, we saw a height increase. We saw a spad meter increase. We saw some other things happening. And if we looked at it, we saw less damage from windstorms from where we had these biostimulants on these crops. We looked at the root systems, and we saw bigger root systems with the yields, with the biostimulants versus the control, and a little bit larger ear size. Some substantial differences, we thought, but when we got to yield, we found that, well, really wasn't much difference. In this case, we had larger stalks, but when we looked at stalk size, again, an increase. When it came to yield, it wasn't there. Reason being, we had a little bit less yield where we had the biostimulants, because I think we had tied up more nutrients in the stalks because we had bigger stalks. We didn't have enough nutrition to bring home what we had set up the plants to do. We've seen some work with jasmonic pathways, the SAR type aspects. We've looked at things on melons here in the Palo Verde Valley. We had a field that had 5 different types of melons, and they were big enough so we could do our plots. Our beds here, or plots were 3 beds wide by field length, and you can see we had some differences in growth early in the season. In the cool, these things covered up the beds a lot faster where they had been treated, and it was true across almost all the varieties. But when it came to marketable melons, it was different by melon variety. Some of the ones that were much less and higher, much reduced in yield, they had lots and lots of foliage. But in this case, there's so much foliage, we think the bees could not get in there to find all the flowers. Kind of an interesting thing that happens, kind of a victim of success. And the thing we've been seeing most recently has been looking at crop stages when these things have applied. We've looked at alfalfa. We've looked at a product called Toggle, which is an extract from a seaweed extract. It's got lots of different things in it, being an extract that way. And it's kind of interesting. If we look at these 4 yields across the board here, when we applied it, at that second or third irrigation, about 14 to 18 days after cutting, we saw a yield increase. The next cutting in July, we put it on right in that first irrigation, no effect. We saw again, we go back to the second or third irrigation in the drip irrigation situation here. Again, I got a tenth of a ton yield increase. And when we go away from not putting it on at all, it tells us the data showed we had no yield. So this kind of indicates that the timing of the application relative to the plant growth in this case is also very critical. And we've seen this also in products that we've used on dehydrator onions. We've looked at these over the years. We've looked at lots of different products. We saw some things several years ago where we had a nice big increase in a sandy soil with a cytopower, different rates compared to the check. So there were some nice differences there. Of course, when we got higher yields, we also kind of reduced some of our solids, which is not to be unexpected, a kind of a dilution factor. All right, so as we look at a comparison of biostimulants, which is really what we want to see here, is how do these things kind of work? So we looked at a bunch of different products on some of these onions. We've got a guaranteed complex on the far left with the purple bars. Those are all, again, that's a seaweed extract, the liquid seaweed concentrate. The green bars is also that. And we got the apenergetic and a check there over on the right. It's kind of interesting that we saw differences by timing. And when we went from the far left, where we've just to put an application on at pre-germination right before we watered it, versus coming on with another application at 3-leaf stage, we dropped those yields. With that particular product coming back at 5th leaf, we brought the yields back up. The green bars over there with the seaweed extract, again, we saw there a combination product of 2 applications was the best. And we saw the same sort of thing again in this past year when we did these experiments again. We have some other things where we looked at onions. We saw some interesting aspects here where we saw a bunch of different products where we looked at things. The far right over there is the untreated. Certain products gave us about a 4 to 6% increase in 2019 and 2020. We did repeat the experiment again in 2020 and 2021, and we saw basically the same response. So we had nice consistency with the same rates and the same timings with different varieties and different growing conditions. So that gave us a lot of confidence that what we're seeing here is solid. So, but one thing we really noticed with this is this application of the biostimulants to crop development stage is extremely important. And not every molecule in these products are going to have the same effect at all growth stages. And we're seeing different responses to different products at different growth stages. So I think if we look at why is success not being consistently realized with all the biostimulants, I think it comes back to remembering that it's easier to increase those growth and yields of biostim, or to increase growth and yields of biostimulants. Everything has to work together, and you have to have the other things in place as well. So I think it's challenging, and I think Nick's going to have lots of fun trying to figure some things out here, as well as all the other regulators, because I think part of our problem to the question is that we're dealing with complex biological systems, and one size does not fit all. Just because one geographic area has stress and a plant responds to the lack of stress or to the stress doesn't mean it's going to happen in another location. And with that, I will take some questions if there's time for questions.
Thank you, Michael. Well, there are no questions at this point. I have a question for you about your experiments. Have you ever tried to apply biostimulants, say, ahead of a heat wave or in response to drought or something like that that would prompt maybe their activity?
We have lots of stress with the onions. We're getting a response here. We don't know if our growth responses we're seeing in the onions are due to a better survival of the heat stress or not. Some of these compounds have betaines in them and other things that are biologically active. We've tried it on cotton with some other products that are not biostimulants, and we've been able to elucidate some effects. But there is such a point as, even with those, that the heat is so much extreme here that we can't even do that. But I think there's lots of spots. But we've tried it with alfalfa, and we're seeing maybe yes and maybe no. Again, each product and each timing is so critical. It's hard to say with a blanket statement what's going to work.
Well, okay, I think in the interest of time, given the delay, we'll end our session today on biostimulants. Thank you, Michael, for an interesting presentation. And I guess Florence may come back on, but if not, I don't know what the schedule is now other than to have lunch. So there's Florence. So I'll let her take it.
Yeah, hi everyone. Well, I guess we have to stick with the schedule now because people might be joining us just for the meeting. So it might be a kind of a short break, or instead of half an hour, it will be 20 minutes. So we'll see you back at 1 p.m. for our business meeting. So we hope you can join us. We are going to elect new members for this coming year, and we are going to present the Student Awards Scholarship and the Lightning Presentation Award. And then also very important, we are going to present the award to our honorees. So please join us back at 1 p.m. for our business meeting. So we'll see you shortly. I guess just 1 o'clock, so let's get started then. Okay, so good afternoon, everyone. I would like to call to order the annual business meeting of the California chapter of the American Society of Agronomy. And in case you are not present yesterday morning, when I introduced myself, just my name is Florence Castle. I'm an associate professor of irrigation and water management at California State University, Fresno. And this year I've been serving as the chapter's president. So I hope you're enjoying the conference so far. We had great presentation yesterday and this morning, and it was also nice to listen to the students who highlighted their research yesterday. They really did a good job. So thank you everyone for attending and also being present today for this afternoon for our business meeting. So before we start the program, I have a few notes to share what we usually do when we are in person at a business meeting. First, I want to mention that the Chapters Conference has been running since 1972, so we take pride in that. This is actually our 51st meeting, so that's a long time. And actually, I want to point out that it's probably our conference is probably one of the longest running conference in California. And actually, by curiosity, I look back over the conference proceeding. You have them all archived. Actually, we have them all archived on our website, so you can take a look at it if you're interested. But that's nice to read some of them. And I noticed that the first conference that we had was in 1972. And so it actually took place the last week of January. And then starting the following year, we had it scheduled from the first week of February. So it's always like consistently, we are scheduling it at the same time. So it has been going on for many years, more than 50 years. So it's really, really great that we are being able to keep with the tradition. So last year also, you see that we have, usually we have proceedings. So we starting calling these proceeding documents actually that you see on the archive really. We started calling it the Book of Abstracts, mostly because we are asking speakers to really just give an abstract versus what we used to do like a proceeding. So now you might see a change in names, but when you go to our website, you should be able to see the Book of Abstract that we put together for this year's conference. So in addition to the abstract, you also find the list of all the board members, the past president of the chapter, past honorees, and also you'll find also the minute of last year's meeting in the appendix. And actually in a few minutes, we are going to vote on that. No? So you might want to take a look. So I would like to also request that you please give us your feedback on the conference. Tomorrow, I think program support, Clean, you're going to post a link to the survey. And if you cannot attend tomorrow, I think it will be emailed to you. Is that correct, Clean?
That's correct.
Yeah. Okay, so we want you to fill out the survey form because we do go through all of these surveys as we prepare for the next conference. And that usually we get some very nice and good suggestions for session topics for next year, future board members and honorees. So that's really important to us. So please do that. Fill out the survey or the evaluation form. That's really helpful to us. Then again, I want to acknowledge and thank our sponsors who generously donated for our student awards. So we had the Western Plant Health Association. So thank you for supporting the student scholarships. Thank you, Renee. Then De La Valley Laboratory, Dan and your team, thank you so much for your continued support. Then we had also donations from Valley Tech Agricultural Laboratory Services, Mid Valley Agricultural Services, Simplot, and Wilbur Ellis. So you have been longtime sponsors of the conference, and so really appreciate it and thank you. So of course, thank you to the ANR program support team. I mean, Clean and Sherry, they've been wonderful working with you. And as I mentioned yesterday, your team has been a great support in planning and organizing the conference this year. We started last year too with your services, and it has been really wonderful. It has taken a big load off our shoulders, really. And last time when as we transition into this virtual environment. That was quite a challenge, but it was very helpful that you were able to help us. I also want to acknowledge and thank the executive committee and the governing board, which really we should call the council of representatives for their hard work in preparing the conference and moderating all the session. So on the executive committee this year, and I don't know, maybe Clean, you can put on the screen the page with all the members. So we had Mark Eddy from CDFA as our secretary treasurer. Then we had Daniel Geisler as our second vice president, and Michelle Landfiler-Mize as our first vice president. And then Eric Ellison was our past president. So there is a lot that goes into behind the scene in preparing for the conference. So thank you for your dedication and for all your many hours of service and volunteering on the executive committee. And then also you see on the sheet we have members who like 9 members on the governing board. So it's a little bit hard to read, but Michael Kahn, Nick Clark, Jacob Wenger, May Colomber, Lauren Hale, Gina Colfer, Ranjit Riar, Sarah Light, and Maria Kovinen. And so they are all serving on the governing board this year. So thank you so much. And I think you have met most of them. They were moderating sessions yesterday and this morning. So thank you all of you for your time and your dedication on the board. So with that, let's start with the first order of business, which is to approve the minutes of last year's business meeting. So if some of you, I think, Clint, if you can show the minutes from last year, yeah, thank you. And also there are, you can find those in our book of abstract. I don't know if you have time to go to that. It will be in the appendix, I think, on page 51. If you go there, you can also see the minutes from our business meeting last year. So I give you maybe a minute to quickly. Can we make it slightly larger, Clint? No, I don't know if it's visible. So in the same way, we had a financial report. We nominated new board members. And then you see listed also the scholarship awards, the student who received the scholarship awards, and the poster awards. So that's mostly the content of our business meeting from last year, the minutes. So if I could get a motion to approve from any member in the audience. Do I have someone? Let's see if I can see.
That one from Sharon Bennett.
Okay, Sharon, move to approve. Okay, thank you, Sharon. Sharon Bennett, move to approve. And then do I have a second? Let's see, I'm trying to monitor the chat and anyone to please, somebody.
Second.
Okay, who was that? Sorry.
That's me, Ranjit.
Oh, Ranjit. Okay, great. Thank you, Ranjit. So thank you, Sharon and Ranjit. Okay, so the last year's minutes have been approved. Thank you, everyone. So now, if we could go back to the agenda. Yeah. Okay, so now number 3 on our agenda is the Treasurer's report, and Mark Eddy, our Secretary Treasurer, is going to present it for us. Mark?
Yes. Yeah, the financial report. So this financial report includes the finances from last year, roughly the calendar year. So the 2021 conference was our first with the program support unit of UC Agricultural and Natural Resources, UC ANR. And so the chapter now has an account with UC ANR that represents a significant part of our assets, in addition to the chapter's Wells Fargo account. Last year, through UCNR, the conference took in a total of $16,820 and incurred $5,943.18 in expenses, leaving us a $10,876.82 in the account. So that's that account. We have so roughly almost 11,000 in the account with UCANR, and then we have our checking account with Wells Fargo, which hardly got any use last year because everything was going through, almost everything was going through ANR except for the scholarships, donations, and awards, student awards, and things like that. And for that year, the beginning balance for the checking account was $32,821.63. The ending balance for the year was $33,518.15. So that's, basically we're pretty comfortable. And I think one of the goals of the chapter is to be able to withstand like a year where we lose all our money or lose all our, the conference has to pay for itself for some reason. And I think we're comfortably in that zone right now. So that's good. We have a healthy organization. As an aside, this is, so sitting in the audience earlier today and yesterday, one thing, a downside, if I can add this to the financial report, of this Zoom online, I mean, we all miss actually seeing each other in 3D, and actually part of that is we don't even know who else is in the room or who else is attending the conference with us. But just to let you know, this year, while you're watching, we have about 100 paid registrants. And in addition to that, quite a few students, especially from Fresno State, because Fresno, some of the agricultural classes have assigned this conference as a requirement for class. So that's a good thing. We let them in free. So we have well over 200 people in attendance. So while you're sitting at home or in your office alone with your screen, you can feel like, well, I'm doing this with lots and lots of other people. I just don't know who exactly they are. So that's the end of my report. If there are any questions, feel free.
Any questions? So actually, normally we need to have a motion to accept this financial report. So can somebody move? Do I have a motion from someone to accept?
Move to accept the report.
Okay, so thank you. I think it was Nick, no? Yeah, Nick. And a second?
I second that motion.
Okay, thank you, Eric. Okay, so the financial report has been approved. Thank you, everyone. Now, number 4 on our agenda, it's related to new members. So, of course, you have seen a little bit, you saw the 9 members that we have on our governing boards, and we have like 5 members on the executive committee. So the executive committee member would serve a 5-year term, starting from secretary treasurer to president and past president, and then the people serving on the Council of Representatives will serve a 3-year term. And then we try to have members that represent various disciplines, and also that come from academia, the industry, and also federal, state, and local agencies. So we have a few members who are completing their term of service this year. So one is actually Eric Ellison. He served as our past president this year, and unfortunately he's going to be leaving the board So Eric served on the board for 8 years as an industry representative and dedicated a lot of time to the chapter. So we relied on him for a lot of things, including a lot of our finances and accounting. And last year he had the difficult task of really transitioning to this online conference, and he did a great job. So thank you, Eric. And actually you're going to see Eric shortly, because he's going to be presenting the Honoris Award segment of our business meeting. Okay, and then we have completing a 3-year term on the governing board. So we have, let's say, Nick Clark, Nick Clark, and then we have Jacob Wenger, and then we had Michael Kahn. So thank you, all of you, for all your 3 years of service. I think you contributed really greatly to the planning of our conference every year, and you have provided a lot of speakers and organized and moderated a lot of sessions. So Michael, particularly, you contributed a lot to our irrigation management session as a session organizer and moderator, and always invited great speakers to address all these current and issues related to irrigation and water, and also with a nice practical application. So thank you, Michael. It was great serving with you. And Jacob, so Jacob Wenger from CSU Fresno. So in addition to your contribution to pest management, as you are chairing for several years the scholarship committee, student scholarship committee, and actually we're going to hear from you very shortly for this year's award. So thank you, Jack. And then we have Nick Clark from UC Cooperative Extension, who she provided us always a lot of inputs to improve our conference. And you have also chaired the student presentation competition for the past few years. And so we are going to see you again in a few minutes as you are going to present the Student Lightning Presentation Award. And Nick is actually going to continue serving the chapter because he's going to join the executive committee as the secretary treasurer. And so that's a 5-year term up to past president. So thank you for agreeing to serve, Nick, and we're happy to have you on the executive committee. So thank you, everyone. So now that we have these people rotating off the board, we need to fill those vacancies. And we have 3 people that who have been nominated by various members of the board, actually from the executive committee and the Council of Representatives. And those 3 people are Ian Gretenberger from UC Davis, Hossein Zakeri from CSU Chico, and Sultan Begna from USDA ARS in Pauline. So thank you. Clean posted their photograph and their bio on the screen. So you can find also those in our book of abstract in the appendices. So quickly, Ian Gretenberger is an assistant specialist in cooperative extension at the University of California, Davis in the Department of Entomology and Nematology. And his work and research and extension work focuses on field and vegetable crop entomology. So he has worked a lot on invasive species, insecticide resistance, biological control, and pest monitoring. So welcome, Ian. I'm not sure, Ian, I'm not sure if you are here. Not too sure if you are able to join, but install maybe, no. Oh yeah, you are on. Do you want to say a few words, Ian?
Oh, yeah, I mean, I think you covered, I think, most of it, but
just
yeah.
so we can,
Yeah, just to
we
say.
can see your face and know who you are.
Yep. Yeah, bring me to the front of the screen. Thanks.
Okay, then our second nominee is Hossein Zakeri, who is an associate professor at CSU Chico, and his work focuses on row crop, and he's teaching at CSU Chico. And if you have come to our in-person conference, you might have seen Hossein bringing a lot of students from Chico State. So it's always good to have that connection with the student and with with another university, and his research really emphasizes on cropping system, diversity, plant nutrition, nitrogen fixation, and so I think Hussein, are you here?
Well, thanks so much. Thank you so much for the nomination and introducing me. I really appreciate that, and I also appreciate the opportunity that this society and the conference has provided for our students. We used to go to this conference, and hopefully we get to go back again with a large group of students to present. It's a great place to be, and I'm happy. Thank you.
Well, thank you for accepting our invitation. Yeah, we look forward to seeing you and your student next year. Hopefully we'll be in person, Yeah. Okay, and then our third nominee is Sultan Begna from USDA ARS in Parlier. Sultan is a research agronomist, specialist in grain and forage crop production, particularly alfalfa. So Sultan worked out of the lab at the USDA in Parlier, and so you worked a lot on forage crops and looking at biotic and abiotic factors. So Sultan, I think you were there before we started.
Yes, yeah. Thank you for the introduction and for this opportunity. I'm very happy to join, your team. It's a very exciting team. This is the second year that I attended, this conference, and the presentation, or the presenters who come to this conference bring very valuable and practical, things that are, necessary for, growers. So I am very excited, happy for this opportunity, and will do my best to contribute as much as I can, to make, not only California, if possible, the whole globe, a better globe for the members of, the world. Thank you.
Okay. Well, thank you so much. So now we need to make these nominations official. And in fact, we need a motion to approve these 3 board members. So if I can please have a motion to approve.
So moved.
Okay, thank you, Ranjit. A second, please.
I second.
Okay, thank you, May. So, okay, so it's good. Officially, all of you are on the governing board now. Congratulations. We look forward to working with you this coming year.
Yeah,
okay,
you.
So yeah, I think so now number 5, I guess, on our agenda is the announcement of the Student Scholarship Award, and Jacob Wenger, who chaired the committee, is going to announce the awardees. So Jack, take it away.
All right, will do. All right, good afternoon, everyone. So every year, the Western Plant Health Association generously donates funds to the California ASA to support an undergraduate scholarship. The CA-ASA undergraduate scholarship aims to elevate the next generation of agronomists by providing financial support to students in agricultural sciences at California universities. This award specifically is targeted at high-achieving students whom have exhibited excellence not only in the classroom, but also in the agricultural industry. So this year, we received some excellent applications and are pleased to announce that 2 students were selected to receive awards, Mr. Aaron Guerra and Mr. Omar Abulghanam. So I will provide a brief summary of Aaron and Omar's accomplishments and then provide them with an opportunity to speak if they wish. So Mr. Aaron Guerra is a 3rd-year student at UC Davis, where he is studying environmental policy analysis and planning. Aaron did not initially envision himself becoming an agronomist, but discovered a passion for soil science and sustainable food production through both his coursework and his employment as a co-coordinator at the Smart Landscape Project in UC Davis's Center for Urban Horticulture. Though the work that Aaron has done has been focused primarily on ornamental horticulture, he hopes to expand into food production in the near future. I don't believe that Aaron is able to make it today. Yeah, I did a search of the chat. He is unfortunately not here. So I will move on to Omar's profile. So Omar is a second-year student at Fresno State University studying plant science. Originally, Omar pursued a career in business administration. However, on a trip to Jordan during the COVID-19 pandemic to help his father, he realized how difficult it is for people in many parts of the world to access clean and fresh produce. This realization inspired Omar to re-enroll in university and pursue a degree in agriculture. Since then, Omar has become highly invested in agriculture, working as a researcher at the USDA ARS, studying whole orchard recycling. And additionally, Omar has been actively participating in Fresno State's Plant Science Club, including travel to the 2021 National Conference of the Tri-Societies, where he participated in soils judging, quiz bowl, and the speech contest, in which he placed fourth. Moving forward, Omar hopes to pursue a career in research, studying topics related to hydrology and soil management practices. I believe that Omar is here on the call, so if he would like to say a few words, that would be very welcome.
Thank you. Can you guys hear me okay?
Yes.
Okay. Thank you, Dr. Wegner. That was a really, really nice words that you had to say about me. I just wanted to come on here and say thank you, especially to the Western Plant Health Association, as well as the American Society of Agronomy for selecting me for this scholarship. It means the world to me as an undergraduate student who has had a lot of his financial obligations fall on himself. Just my dedication to my academics and my continuation to work hard. This is just a testament that you work hard and good things happen. So thank you to the American Society of Agronomy, as well as CSU Fresno. California State University Fresno has supported me in so many ways. Their professors that they have there really inspire their students through the research that they do, as well as the ideas that kind of just flow through the plant science department there. So special thank you to Dr. Sharon Bennis and Dr. Ranjit Riar for all the support that they've given me, as well as here at the USDA. I would like to give a thank you to Dr. Sudawan Gao for bringing me onto her research team and showing me and teaching me and supporting me in everything. So I look forward to my future, my academics, and continuing to explore the new sciences that are emerging in agriculture in California. So thank you to everybody. Thank you.
Excellent. Thank you very much, Omar.
Yeah, thank you, Omar. Congratulations. And thank you, Jack, for this presentation. Okay, so next, number 6 on our agenda, it's time to announce the Student Lightning Presentation Awards, and Nick Clark, who chaired the committee, will be presenting. So, Nick.
Thanks, Florence, and good afternoon again, everybody. I'm honored to be able to present the awards for placing first, second, and third in the second Student Lightning Talk competition that we've conducted at this conference. But before that, I want to just extend a thank you to the presenters yesterday. Of course, thank you again to the judges that judged those talks, Gina Colfer, Daniel Geisler, and Michael Kahn. The co-chairs for this session were Mark Cady and with CDFA and Ranjit Riar with CSU Fresno, who really kept kept me on track in organizing this session. Thank you for that. Thank you for all of the attendees yesterday that supported the students during this session and asked good questions of them to keep them on their toes and interested in this profession. And last but not least, of course, thank you to the sponsors that help us give out these awards. So the process that I have for announcing the winners of the competition is I'll announce the names of the presenters, the title of their presentation, and what the cash award amount is that they'll be receiving. And then after I've announced the winners, I'll give each of them if they're present, a chance to say a few words if they choose. So without further ado, I'd like to announce that our first place winner of this year's Student Lightning Talk competition is Samantha Jackson. Her presentation was called The Proportion of Belbean, Fava bean Biomass and Nitrogen in Cover Crop Mixes in Annual Crops and Orchards in Northern California. For first place, Samantha will be awarded $600. Our second place winner for the competition is Lauren Marigliano, whose presentation was titled Photoselective Shade Films Affect Grapevine Berry Secondary Metabolism and Wine Composition. And for that, for placing second, Lauren will take home a $500 award. And finally, our third place winner for the competition was Aaron Guerra for his presentation titled Smart Lawn Demonstration, Water Budget Calculations for Warm Season Turfgrass and Turfgrass Alternative to Meet California's Landscape Ordinance. For winning third place, Aaron will take home $400 as an award. Congratulations to all three of the presenters for placing. And I think at this moment, Samantha Jackson might be present and able to chat. Or Samantha, would you like to say a few words either live or through the chat?
Yeah, I'm here. Yeah, I just really want to say thank you. A huge thank you to Dr. Zachary. I would not have any of the opportunities that I've had without him. I never would have seen myself in research at all without Dr. Zachary or gotten where I am today. So just a big thank you, and just all the support out at the CSU Chico Farm and the College of Ag. So it's just been such an awesome experience. So thank you so much to everyone.
Congratulations, and Lauren Marigliano, if you're with us and you'd like to say a few words, you're welcome.
Here. Thank you so much to the sponsors for supporting these kind of talks. They're always really great practice for, sharing our research and having public speaking skills. And I'm really interested in going into extension. So being able to participate in these types of things is really is a lot of fun for me. So thank you.
Thank you very much. And I don't believe that Aaron Guerra is with us in attendance right now. So that concludes the announcement of the Student Lightning Talk Competition Awards. Again, congratulations to all of the students on your accomplishments, and we look forward to your contributions to the profession in years to come.
Thank you to all the students, and thank you, Nick, for presenting these awards. Also, I think we have a little bit of time, but I want to say that other students also chose to submit a poster to share their work and research. So you can go on our website and you see the poster gallery on our landing page. So please also visit it and look at the work that other students have done as well. So thank you so much to the committee for organizing these presentations. So now I think we're a little bit ahead of schedule based on what's written in our agenda, but I guess Eric is going to be presenting the honorees and the awards. And I guess that will give you a little bit of extra time then, Eric. It's not really too bad.
That's great. Well, thank you everyone for attending this business meeting. I just wanted to remind everyone that sometimes we forget that everyone who attends this meeting is a member of CA-ASA, and you have voting rights, and I think, you know, your participation and your input is appreciated. As past president of the chapter, one of my roles on the board this year is to chair the honoree session, and I feel privileged to do so this year. Each year, CA-ASA honors professionals who have made outstanding contributions in advancing California agriculture through their research, through dissemination of scientific information, teaching, and collaboration. In most years, those chosen to be honored are retired from their profession. At least they have moved on from their career to work other jobs, maybe as consultants, or maybe they just want to solve Wordle puzzles. I don't know. You know, I know a lot of our honorees are still very productive, even though they may be so-called retired. And many of the honorees have served the CA-ASA governing board. Nominations for the honorary award are drawn from conference evaluations and suggestions from board members. And that's where your participation as a member of CA-ASA is important. You can always suggest a person to be nominated or considered as an honoree, and we appreciate that. So please continue to do that. We have used that as a valuable resource. Deciding who to honor each year is never easy because there's so many accomplished professionals working in California agriculture. And this year was no different. On behalf of the governing board, we're pleased to select as the 2022 CA-ASA honorees two very deserving, very kind-hearted and friendly individuals. Bruce Roberts, emeritus faculty member with Fresno State University Plant Sciences Department, and Blake Sandin, farm advisor emeritus with University of California Cooperative Extension in Kern County. I encourage everyone to take time to read the biographies for Bruce and Blake that were published in the Book of Abstracts. And for students in particular, take note of the humble beginnings. All of us as scientists, we all start somewhere, and it's the big unknown, and sometimes we think we know. Maybe we think we know more than we actually do when we're younger, and then as we grow older, obviously we begin to realize how much we don't know. And that's part of the challenge, and that's part of the excitement. But we can ask ourselves, or you can ask yourself, what drives someone to work long hours, like Blake plumbing a pistachio orchard to determine how much water a tree really needs, or like Bruce to understand potassium fixation in soils and how it affects cotton growth and yield? I can't really speak for either Bruce or Blake, but I'm sure the answer is probably based in a strong work ethic and that they were driven to solve important problems. They accepted big challenges, probably where they thought they could make a difference. And they did make a difference. And their reward was being able to communicate new and useful information that helped a lot of people, like a pistachio or a cotton grower. And I'm sure they gained a lot of job satisfaction from doing that. And sometimes I say this in jest, the fool who rushes in gets the job done. And I think as a scientist, sometimes we just have to take on these jobs, even though we don't know necessarily how they're going to turn out. But we just, as a scientist, you've got to do something. But let's all congratulate Bruce and Blake for their many accomplishments, of course, in silence, because most of you are muted, but you can use the chat bar to make comments or whatever you want to do. Now, in keeping with tradition, each honoree will be introduced by a colleague of their choice. We will begin with the introduction of Professor Roberts. Bruce will be introduced by Philip Smith. He's area soil scientist with the National Resource Conservation Service in Salinas. And again, I'll point out that attendees who are not on the panel can send comments to each honoree by using the chat bar, and feel free to do that. So Phil, go ahead with the introduction of Bruce Roberts. Thank you.
Thank you, Eric. It's good to be here today with you all. As Eric said, I'm Phil Smith. I work for USDA NRCS over in Salinas, where I'm the area soil scientist on the Central Coast. But before I moved to Salinas last year, I lived in Hanford, and I worked a lot at Central Valley. And that's when I got to know Dr. Bruce Roberts, whom I have the honor to introduce and present today here at this meeting. So, as a matter of fact, it was at this meeting 8 years ago when I met Bruce for the very first time. I was standing out in the foyer of the conference center there in Fresno, standing out there with some folks, and that's when I started talking with Dr. Roberts right there for the very first time at this meeting, and got to know him over the years and consider him a good friend and person that I'm glad that I ran into during my stint over in the Central Valley. This morning, when I was I talked with Bruce this morning to think about what I'm going to say today in this introduction, and one thing Bruce told me was he talked about the importance of the California chapter and this meeting, this conference, the American Society of Agronomy, Soil Science Society of America, but especially the California group, because it's important. And he, the science is one thing, the agronomy, that's one thing, but and then the people is another thing. But here's a quote. Bruce just told me this, and I said, let me write that down. Let me see, that's a quote. He said, "It's the people you get a chance to be with. It's the people you get to meet and have a cup of coffee with. That's what matters. It's the fellowship of getting to know your colleagues, and this is very important. That's why the meetings are important." And that's what Bruce told me this morning. Again, I met him at this meeting, having coffee out in the foyer, and maybe next year we can get back to having these meetings in person, because that's one of the great things, where we all can, this meeting brings together people from the universities, extension, consultants, USDA, students, people from all over California who work in agriculture. So some information about Bruce. So Bruce grew up in Chowchilla, in the heart of Central California agriculture. And his father had about a 40-acre irrigated farm, and they also did some dry land farming. He had about half of the irrigated land was in Alberta peaches. The other half was in cotton, and that, later on, that became an important part of Bruce's career, cotton. A few things you may not know about Bruce. He was drafted in the Army. He went to the Army in 1969 and did 2 years in the Army in Alaska. He received an associate's degree from Fresno City College. Then he went up to Humboldt State, and that's where he took his first course in natural resources conservation. But then to get closer back to his the love of his life, Amy, he transferred down to Fresno State, where he got his bachelor's degree in the plant science department. And, eventually he would make his way back to the plant science department years later. A quote from Antoine Saint-Exupéry: "If one believes education is an end, then he does not understand the meaning of means and ends." In 1975, Bruce defined his career objective and formed a hypothesis. and the reason for applying to the graduate program at Utah State University. He believed that a career in cooperative extension would be an excellent foundation to prepare one for college teaching. That was his hypothesis. Now, after graduating from Utah State with a master's degree in soil plant water relations, Bruce came back to California, moved over to King City in Monterey County, where he worked for Basic Vegetables. And there he did research and field trials for them all over the state, really from Imperial County to Monterey County and probably everywhere in between. In 1983, Bruce started as a farm advisor in Kings County and began his career in cooperative extension. I want to say a few things about Bruce's time in Kings County. And this is in the program write-up. So during the 2 decades Bruce served as a farm advisor in Kings County, California experienced 2 major floods and 2 long droughts. These were exceptionally challenging times for California farmers, especially for growers in the Tulare Lake bed. Flooding disrupted crop rotations, and late spring rains caused delayed plantings and outbreaks of seedling diseases. The droughts forced researchers and growers to explore means of conserving irrigation water, practices and limitations that have continued to challenge California farmers. The extension program Bruce conducted in Kings County addressed many production constraints related to agronomic crops produced in the San Joaquin Valley. Applied field research and extension outreach meetings addressed issues of seedling disease, soil fertility, variety evaluations, pest management practices, pre-harvest preparations, and storage of module seed cotton. Evidence of Bruce's extension efforts are reflected in changes of cotton lint yields over those 20 years. Average lint yields reported in Kings County rose from 2 bales in 1983 to 3 bales per acre in 2004, a 47% increase in cotton lint yields across the county. And by 2004, several growers reported 4 bale per acre yields from new varieties and careful, better management practices. Bruce's extension program also involved the introduction of new crops. Garbanzo beans was introduced as a viable alternative to winter cereals, and Kings County played a significant role in the acceptance and approval of long staple Pima cotton for the San Joaquin Valley. So, you know, all that didn't just happen. It took important work from people like Bruce Roberts to make this transform agriculture in Kings County, and that is where Bruce spent about 20 years of his career at serving the people in Kings County. He also conducted entomology field trials and chamber studies. He's proud of the collaborative work he did with the other scientists, and some of those studies were potassium fertility studies that he did with Ken Castman, an energy balance study on irrigated cotton. He collaborated with NOAA scientists, the National Oceanic and Atmospheric Administration. He also conducted nitrogen defoliation studies on cotton, and in sharing his knowledge, in sharing his research, the precision ag field days, the field days where you share your knowledge in Kings County, these drew over 300 participants. And I encourage you to read the write-up that's in the program. I'm just touching on a few things that Bruce did in extension. In 1998, he was able to take a sabbatical and attend classes at UC Davis, where he pursued his PhD. He was accepted into the graduate group of ecology, and his dissertation delved into the domain of soil ecology using microbiology and chemistry. He partitioned and quantitatively measured the decomposition products produced by distinct microbial communities. Now, okay, so now following his year at UC Davis, Bruce got to test his hypothesis. So he realized the opportunity to make that transition from extension service to being a college professor and being a teacher at Fresno State. The J.G. Boswell Chair of Agronomy was created at Fresno State, and Bruce applied for that, and he became the first person to hold that position in the plant science department. And it's at the plant science department at Fresno State that I got to know Bruce personally, and like I said, it was in 2014 at this meeting, and since then I've had the opportunity to. It was really with Bruce that Bruce opened the door for me to work with the plant science department at Fresno State, and share soil science with the students there. But that was really started through my conversations with Bruce. But Bruce was an outstanding teacher at Fresno State, and just his commitment to the students. I noticed that right up front when I first started getting to know Bruce. He encouraged students to be members of the plant science club. They reinstated that during Bruce's tenure there. He always encouraged students to attend the national meetings. An example. I remember, I think we were at the Tri-Societies meetings. It was Long Beach, I think is where it was. And I just, by chance, ran into Dr. Roberts and a whole lot of Fresno State students. And Fresno State always represented very well at these national meetings. They would always bring many students, I don't know, probably 10, 15 sometimes. So way more than other universities, especially with undergraduate students. They're well represented, and Bruce can be recognized for that. He helped form the soil judging team, and that was an opportunity for me to work with the plant science department. But that was really Bruce was there at the very beginning with that, and he developed ties with industry to support student involvement and travel. I've seen few people so gifted at working with industry and professionals and people in the agricultural economy and getting them to support the students the way that Dr. Roberts did. In 2010, Bruce was selected as a fellow for the American Society of Agronomy, and in 2015 he received the Salgo Noren Outstanding Teacher Award at Fresno State. Bruce is honored to be a faculty advisor of Alpha Gamma Rho, a role that he had held for 10 years. So back to the Bruce's hypothesis. Bruce spent the last decade of his career testing his early hypothesis that an extension career would be a beneficial foundation to prepare one for college teaching. It turned out to be an excellent foundation for training the future generation of agricultural producers, scientists, and lifelong learners for California. Bruce is honored to have known and worked with such a brilliant and dedicated group of scientists, farmers, students over his career in extension and teaching. Bruce is a past president of this chapter, the California American Society of Agronomy. That was in 2005. And he served previously on the board of directors. His research has been presented in book chapters, scientific publications, and at international symposiums, national conferences, statewide professional meetings. He retired in 2016 and turned his interest to pursuits he's always wanted to do. And these include traveling to exotic places, painting with watercolors, and these are some of his new goals that he has done in retirement, including he plans to paint his masterpiece, watercolors. So, I mean, who knew that Bruce did watercolor paintings? A few other things I want to mention. In 2010, before his 61st birthday, how many of you knew this? He spent 4 days climbing the nose route on Yosemite's El Capitan. Wow. That's quite a lifetime accomplishment right there. Climbing El Capitan. Way to go, Bruce. He and his wife Amy have also rode the Grand Canyon's Colorado River for their honeymoon, and married to Amy for 48 years. Together, they have 3 adult children, and being a father has brought. Bruce says that being a father has brought him the most satisfaction and joy of his life. So, well, it is, like I said earlier, I've known Bruce for about 8 years from this meeting, and we've had a great relationship. And today it's my honor to do this, to present this for Bruce. So Bruce has it's already been mailed to him. I believe Bruce has this. And so Bruce, do you want to show us your award plaque? All right, there we go. And I'll let you say a few words.
Thank you. Thank you, Phil. This is quite an honor, and I just want to start off by thanking you for your kind words and what you've done for us. But I also want to thank the board of directors and for their vote of confidence in the selection. I want to congratulate Florence for her new presidency, and congratulate Eric for his past service. And appreciate all from the board of directors, all of their support, because I know what this volunteer board does, the activity and the work that they're involved with of promoting this chapter in our state professionals. So thank you to all the board. I also want to send an acknowledgement out to all my friends and farmers in Kings County who for 20 years allowed me to exercise my curiosity in their fields and hospitality in their homes, and in helping us do that. And want to appreciate everything I've done, my extension colleagues that and the people that I've worked with over the past, because it's always been a team effort, and being part of it, and this chapter involvement of that has always been a very important role. And I just want to speak, if I can, just a few minutes to the younger members of our audience, hopefully students who have joined us. Lauren and Omar, congratulations on your awards and scholarships. If you look in in the write-ups, you'll see that your story and your desires is being played out all the time by people who get inspired by some type of recognition or a life-changing experience that really makes them aware of how important agriculture is to our society and things. It's a wonderful career. It's been a great ride. I just want to share to our younger members reference to Ecclesiastes 1:9, saying that there's really nothing new under the sun. And if you look at a lot of the topics that have been discussed today and things that I've done in research or anybody, don't feel inhibited by if something's done. Don't worry about, take chance of questioning the common knowledge. Just like Blake's shown that there's better improvements of water management in almonds that was against the grain or established farmer. Everything has been done, and we can re-examine those same questions and those problems with today's technology, new tools of DNA, like what Ann Hirsch talked about, about finding bacterial strains that are important and things. It's just a new era for us in agriculture, and this younger generation are going to be key players in doing this and gain from your education and gain from your experience and work towards this. I do want to just use a quote from my son, that's an economist with the USDA in Washington, D.C., sent me this morning. He says, "I'm also lucky you took me to work with you and showed me what it is to really work hard and get into the mud to study something that is useful and important." And agriculture is useful and important to our world. And those young people out there, boy, I wish I could, no, I'm retired. I don't want to do it over again. But I just really think that you've got an exciting career for you ahead of you, and get involved, get active with this chapter and attend these meetings and stay in touch with what's going on, because you're going to be faced with some significant challenges that we can't even imagine right now. Anyway, I just want to express my appreciation to all of the members and friends in this agricultural society in our community for this recognition. And I just love you all. And anyway, that's all I can say is I'm just very touched. Thank you. Back to you, Eric.
Wonderful, Bruce. Congratulations again. And thank you, Phil, for a great introduction. So our next honoree is Blake Sandin, and he will be introduced by Bob Beatty, Farm Advisor Emeritus with UC Cooperative Extension in Kings County. So Bob?
Thank you, Eric, and I assume you can hear me all? Okay. Well, this has been a bit like Old Home Week, because before I start with Blake, I want to extend my sincere thanks for Bruce Roberts. I know him as Bruce because he contributed so significantly to the University of California Cooperative Extension Office in Kings County for his 20 years here. And between Bruce and Alan Fulton and Carol Collar and myself and Joe Padilla, why we were a small county, but we were mighty in the contributions that we made. And I'll still see Ken Casman walking the halls with Bruce. We used to call him Gum Chewing Ken because we never saw him without gum in his mouth. And it was during those times that they discovered this potassium wilt in Kings County and made just huge advancements. And so all the blessings and honor go to Bruce. Oh, as well as Dr. Roberts. I wanted to be sure that you knew that the Carl Finucci family extended their sincere appreciation to you and the honors that you definitely deserve. You have run the race, Bruce, and we are eternally grateful for all the contributions you not only made to the University of California, but also through Fresno State University. You have continued your passion for extending to young people the importance that agriculture plays in America. And I'm hoping that these 80 participants that are here listening to these talks include many of those young people, because this is not just an opportunity to pat us old farts on the back and try to remember our accolades, but to use this as an opportunity to show the young people that through this arduous journey that one must travel in life, that you have an opportunity to make a true impact, not only in our society, but especially to contribute to the welfare of others. And Blake Sandin is certainly one of those individuals. I have the pleasure of meeting Blake at UC Davis back in the early 70s when Blake got his agronomy degree in 1978. At Davis, he met the love of his life, Sue McCullough, and she also was going to Davis and got a degree in genetics and became a very successful teacher in several locations. And together, after they were had gotten their bachelor's degrees, they pursued missionary service in Zambia for 3.5 years. And as it has been described in the write-up, why they were involved in many aspects of improving the well-being of individuals that do not have the pleasure of the wonderful societal structure that we have in the United States of America. And we are counting on the young people to preserve the integrity of our nation, not only through their efforts in agriculture, but in their personal behavior and their commitment to the American way. And from there, he returned and went to work for in the small community of Woodlake. But shortly after his presence there for a year or two, he then returned to Davis to get his master's degree in 1987. And at that point then got a job with the old Leland McCarthy Ranch. And for those of you that know Westside agriculture, you certainly appreciate the role that Leland McCarthy played, as well as our former county director and former Kern County farm advisor, George Ferry, who helped develop the Westside into the entity that it is today. And Blake served as the irrigation manager during his time at what was then Prudential when Leland sold it to the insurance company, and served as the irrigation supervisor and manager of thousands of acres of crops that were developing. And just so that the young people know who are now enjoying the fruits of low volume irrigation systems, when I first came to Kings County in 1979 and was asked to go out to take a look at the pistachios with Gary Weinberger, and on one of those occasions, I took Dr. David Goldhammer with me. We stood out in the fields and watched the workers irrigate half-mile runs of young pistachio trees that had a little furrow on either side. And this was not flat ground. This ground had a slope to it. And because of the slope, the workers were putting small pontoons into these furrows to block the water on half-mile irrigation runs. And it was at that time that Dr. Goldhammer looked at me and said, "Oh, we can do better than this." And that is the theme that Bruce has shared with you, young people, and I wish to share with you also. There is a new and better way in the future. And Blake certainly demonstrated that when we welcomed him into the University of California staff as a staff member at in Kern County, I believe in 1988. And even though he got a late start as a farm advisor, he most definitely burned the streets up with his enthusiasm and his arduous work effort. Blake, you have done a phenomenal job in contributing not only to the research database of the perennial crops as well as alfalfa and citrus, but also to extending that to growers throughout our southern San Joaquin Valley and the state. And as you all know, Kern County, those of us in Extension always kind of saw Kern County as an entity or a state unto itself. because in Kern County, there were big ideas, big growers, and these were shakers and movers. And you can see that even in their early efforts to bank water, recognizing that although Kern County was somewhat parched in rainfall, that the magnitude of agriculture was so great that it required a lot of water. And so they initiated banking procedures. Blake's research is extensive. In support, in just these past 13 years of his tenure, during UC tenure, Blake was a principal investigator on 18 projects. He co-PI'd 9 and was a cooperator on 9. Many of these projects attendees involved huge commitments of time and labor from both himself and those that assisted him, installing miles, literally miles. In one project, Blake installed 310 miles of subsurface pipe in order to achieve the objective. And he worked with farmers that after they discovered the successes that Blake was seeing in his irrigation projects, that they wanted to change. They didn't want to participate anymore because they'd already seen how great the successes were. And so Blake then had to negotiate with people like Mr. Starr as to how he was going to continue the irrigation regimes in his pistachio project and provide the industry with the needed data that we needed on salinity management for the new rootstocks. And he successfully did that, although with great personal effort. In some of his irrigation water use efficiency studies, he continuously recorded soil moisture sensors in 145 fields covering more than 12,000 acres, belonging to 33 different growers in 14 different crops. And in so doing, increased the yield. And these projects were cited by the Department of Water Resources and one of the largest civil engineering contractors in the San Joaquin Valley in their policy development for irrigated lands regulatory reform. As Bruce has mentioned, he was instrumental in the development of water use and yield, where he monitored 34 almond blocks with greater than 70% canopy and 90% water use efficiency. And in this project, discovered that they were using far more water than what they anticipated. And from that project, he then launched another 5-year project that was statewide, where they looked at 70 to 100% and 110% of ET. And from that, the crop coefficients and water use data was refined dramatically. He also, as I mentioned, worked extensively in pistachios, not only looking at salinity, but also joined forces with Dr. Danielle Zachariah and Julia Moreno, now specialist at Kearney, in examining the impact the salinity had on the shaded area and subsequent reduction in water use. And this data is now used today. Blake is an outstanding teacher. His teaching efforts represent over 200 extension presentations in the past 13 years. And Blake has been just an outstanding individual. He's not only driven, but one of the things that I truly appreciate about Blake is his willingness to listen. Those of you, we have to kid Blake a little bit, that those of you that might have a question that called Blake, why, you better have a tall cup of coffee to hear his answer, because he wasn't going to give you the short version. It was his opportunity to extend to you the information that he had learned in hope that you could then assimilate that and carry the ball to the goal line. And so Blake, even though there could be many more things said about you, I have to say that in conclusion, given all the outstanding research and knowledge you have contributed to the University of California and California agriculture. I am truly honored to be the one blessed with the opportunity to share your career with the American Society of Agronomy and everyone else who recognizes the significance of your contributions to precision irrigation and soil management. I join the Society members today in honoring you for your distinguished service to the Society and to California farmers responsible for ensuring a safe, abundant food supply. Congratulations, my brother Blake.
Thanks, Bob. I have to say that I was very much by the CA-ASA chapter offering me this honor. I have so much enjoyed my participation with the group over a lot of years. I'm, close to 20 years, 30 years. And of course, Bob and I go back 40 years now. And to those of you young people that are so important to this industry going forward, I continue the encouragement that Bruce offered in saying, some of us old dogs had the opportunity to really pick off some great low-hanging fruit. The potassium nutrition in cotton, potassium nutrition in almonds, understanding improved water management in almonds, almost doubling the statewide average California yield. I feel very privileged to have been part of that. But if you read the bio that Bob wrote up and what he just said, basically, I was just a plumber that had the benefit of being able to work with a lot of smart people that did really good things. As California made the transition from where cotton was king up through the late 80s to where now we produce by far and away the majority of fruit and nut crops that are sold throughout the whole nation. Almonds alone are a bigger acreage right now than cotton or alfalfa ever were. So I just want to express my gratitude to the California ASA chapter and to say that as somebody who was really only thinking about agronomy, field crops, grain, what do people need to eat, and having the chance to morph that into an understanding of how you manage almonds and pistachios in terms of water salinity management. I just feel very humbled with this honor, and I commend Bruce for his decades of contribution to ag as we change from the land where cotton is king to the world's greatest almond producer. So, what challenges lie ahead to the rest of you young people attending this meeting, only God knows. And those challenges are big. We've got significant water reductions. We've got the last 20 years of just nasty drought tendencies. So I want to encourage you to go forward. And again, thank you, CA-ASA, for honoring me. And God bless us all down the road. Thank you.
Thank you, Blake, and congratulations to you again as well. And I just want to say that it seems a common trait between both Bruce and Blake is being humble. And I think that really goes a long way in being that's a virtue that we should all strive for. And anyway, again, thank you all for great presentations. It was wonderful. And I'll turn it over to Florence.
Yeah, thank you, Eric. And again, congratulations to you, Bruce and Blake. Really, really well deserved. Bruce, it was great being as presented with you. So thank you for all you have done with the students in the department. And then Blake, it's since I've been coming to this conference, it was always great listening to your presentation. You're so passionate when you present. I mean, it's really inspiring. So, and then I have to say, when we met at this symposium, it was really great last December to reconnect and to see you in person. So, congratulations. Okay, so now let's move on to item 8 on our agenda, which is additional discussion as requested by the membership. Any item or discussion that anyone would like to add? If you please could come forward. Anything you would want to say as part of the meeting? Now let's see, I'm trying to monitor the chat and the Q&A. Anything to add for now? Okay, so you always have the opportunity, you know, when we are going to put forward this the survey, the evaluation. So if you have any comments, suggestions, questions, just feel free to add those. And when you fill out the evaluation, as we said, it's really valuable for us as we try to organize and plan the next conference. Okay, so now the most important part of this meeting is the passing of the gavel to our incoming president, who is going to be Michel Landfinder-Miles. And actually I have a little story before I officially present you the gavel, Michel, is we always do that as a tradition at the meeting. Now it's really hard for you to see, but we have this gavel, and actually Michel and we talked about, okay, how do I pass this gavel along to Michel, and I was going to mail it to her, but then we decided that maybe it's probably safer to wait that we meet in person, and hopefully we can do that in the very near future, because we are always like really careful about what we do with this gavel. We get it in a box, and it has been custom made for the chapter. I think it was made in 1978, and it's made of a lot of different woods, and it's hard to see, but the handle has a lot of different woods from California, and then the head is mostly from different parts of the US, including from Washington, DC. So it's really something that we want to cherish and keep really carefully in our possession. So now with that, Michel, I'm officially kind of passing virtually. Hopefully we could do that in person, but I'm officially passing you the gavel. So you are officially the president of our chapter now. Michel, we are really happy that we are taking on this role. We are really in good hands. And thank you so much for all your services over the year. And I wish you all the best for this year. It's a lot of work, but it's always good. So Michel, are you on? Or?
I'm on. Thank you, Florence. And for the audience, Florence and I had conversations about the gavel and how to go about transferring the gavel. And I think I was secretly, though I never said this to her, wounded because I learned of Christmas cards that I had sent not arriving until January. And so I thought, let's not mail a gavel.
Yeah.
COVID has kind of changed the way that we run the organization, but one thing is remains steady is that the people on the board are very dedicated to this group and seeing this conference be successful. And Florence has certainly pulled off a very successful conference. The one thing that the audience won't know is that the board had a difficult decision to make Earlier in 2021, when we were meeting as a group, we were ready to bring this meeting back in person in Fresno again, like we've done for so many years. And we were making plans to do that. And then as the COVID pandemic was continuing to be a problem, we were faced with the question of whether to be in person or whether to keep it a virtual conference again. And of course, we know there's pros and cons to doing both things. And certainly one of the cons to being virtual is that we don't all get to be together and network the way we enjoy doing. But Florence, as the president, is the one who really has to pull it off. And she's done it very successfully. And we're all very grateful for her leadership this year. So thank you, Florence, for seeing us through another virtual conference and for leading the organization again.
Oh, thank you so much, Michelle. It has been great, really, serving on the board. And as you were saying, we're doing all this meeting virtually, but I kind of, I'm happy that we had those meetings because at least we could connect during this time when for the last 2-3 years, or 2 years, we haven't seen each other that much. And we used to see each other at conferences or at meetings. And so it was kind of nice and refreshing to see everyone on the board. And yeah, we kind of were wondering at some time, what are we going to do, which format? And we really wanted to bring this conference back in person, but I think for everybody, it was probably the best option. But I mean, it has been great serving with everyone on the executive committee and with the board in general. And I mean, it's not so. I was the president, but you can't do it all alone. So I think it takes a team to come up with all the speakers, the session, and all these little things that you have to do in the background that unless you're on the executive committee, you don't really realize. And Michelle, you have a preview of that already. Yeah, it's thank you for all of your help and support for everyone on the board. It has been great, and I really enjoyed those years. So thank you to everyone. And I have to say that, yeah, usually we get a plaque now, so I think I got that plaque. So thank you so much too for that.
Well, and as Florence knows, and as Eric can attest to, and if Karen Lowell had been able to join us this year, she would attest to it as well. The president is not really off the hook. The president continues to serve as past president and then as a council basically for years to come. And so I know that we'll try to release you of some responsibilities, but we're glad that you're around again as past president to provide additional guidance for us.
Oh, yeah, well, sure. And of course, and I did the same thing with Eric. I think I bugged Eric quite a bit this year with all his knowledge about what to do and not to do. So yeah, sure, I'll be happy for that. And you have helped also so much this year with this conference. So I know we are definitely in good hands. So thank you so much, Michelle.
You bet.
It's nice to see everyone again.
And I guess then it's my responsibility to solicit a motion to adjourn the business meeting. Obviously, this is not the end of the conference. We still have some more sessions tomorrow, but we will adjourn the business meeting with a motion. Can I receive a motion from the audience?
I will motion it, Sarah Light.
All right. Sarah has motioned for adjournment. Do I have a second?
I second. Daniel Geisler.
Daniel Geisler seconds. And Sharon Venice seconds. Seconds and thirds. Thank you, everybody, for your participation today in the business meeting. And at 2:30, exactly right on time, we have adjourned our 2022 business meeting of the Plant and Soil Conference of the California chapter of the American Society of Agronomy. Thank you for attending, and we'll see you tomorrow.